learning about light both interesting and fun. The exhibits in the Light Exploratorium will be constantly upgraded and modified so that people visiting it after a while would have something new to learn about light.
A very good morning to one and all, honorable dignitaries, distinguished faculties, seniors, and my dear fellow mates. I heartily welcome you all to the day 2 of theme meeting in life sciences. So, with an objective of encouraging the spirit of research and innovation, let us begin with session 4, which comprises of 4 invited talks today. It will be my pleasure to invite Professor Sunil Ghosh, Associate Dean of Life Sciences, HBNI. So before I invite him onto the stage, I would like to give a brief introduction of him. So Dr. Ghosh is currently DAE Rajaramana Fellow and Associate Dean at HBNI. He did his MSc in 1984 and PhD in 1989 from IIT Kharagpur. He did his postdoctoral research in Cambridge University, UK from 1991 to 93 and in University of Illinois, USA 1999 to 2001. Dr. Ghosh is the product of 30th batch of the prestigious BARC training school. Following successful completion of 1 year orientation course, he joined the bioorganic division of BARC in 1987. He was the head of the Food Technology Division of BARC from 2016 to 20. He became the Associate Group Director of the Bioscience Group of BARC in 2020. Dr. Ghosh has mentored about 16 PhD students and he has over 140 publications in peer-reviewed journals. His research expertise has been the synthetic organic and organometallic chemistry, food chemistry, and radiation processing of food products. Dr. Ghosh is the recipient of several awards like Indian National Science Academy Young Scientist Award in 1992, Homi Bhabha Science and Technology Award 1987, among others. He is the fellow of Maharashtra Academy of Sciences. So, Dr. Ghosh, I welcome you onto the stage and I hand over the mic to you for ahead, proceeding the session.
So a very good morning, and I welcome you all for the session 4. So in the session 4, there are 4 speakers. The first speaker is Professor Prasanna Venkataraman. She is Deputy Director at TMC ACTREC. She is also convener of the Board of Studies Life Sciences, and her research interests are that include protein interactions, network, cancer therapeutic targets, and drugs. The title of her presentation is An Algorithm Integrating Structure and Mass Spectroscopy Derived High Throughput APMS Data Defines Novel Biological Role of a Proteosomal Assembly Chaperone. Now I invite Dr. Prasanna to deliver her lecture. Prasanna Madam. Madam, the lecture times will be for 25 minutes and then 5 minutes we'll keep for discussion.
You have 25 minutes.
Thank you.
That's good. Start karo. Hello. That's a point. Good morning, everyone. And special thanks to Professor Vasudeva Rao, Professor Maiti, and Professor Naik, and Professor Ghosh for conceiving this idea of focus meeting on life sciences. The 4 of them have such an enthusiasm that it is infectious. And I thank all of RRCAT for organizing this meeting here and for an excellent hospitality. They whisked me away for breakfast after I came here, just to make sure that I don't give a talk on an empty stomach. So don't get worried about the title. I think I made it with some specific intention, but then I have tweaked the story to cover most of the audience interest. And don't worry about the number of slides also. They'll get skipped. So my lab is interested in protein interactions. And protein interactions, I probably don't have to emphasize after talk by Abhijit. But nevertheless, they are basis of which the cellular structure and functions are built. You have DNA, RNA, and then the proteins. But when proteins communicate with each other, that's when things happen. Protein interaction act as regulatory nodes in many cellular signaling networks, and that are linked to hallmarks of cancer. They are writers, they are readers, they are erasers, they are executioners, they do multiple functions. And there are also context-dependent interaction. Within a cell, an interaction would look like this, right? It's everybody talks to everybody else, and you don't know how to tweak these to find out what may be important or what may be important in a particular context. Now, this is something similar to the social network. You can look at it 2 ways. there is one important person to whom many people are connected. And you can also see that there are 3 people, and because of their independent connection, the whole network is established. Similar to that, proteins have players who are central to a particular network. There are master players who have multiple interactions. There are proteins which can have such localized network. And because of this, all of the proteins talk to each other. Now, there are many ways of looking at protein interaction depending upon which scientist it is and what their interests are. I was trained in looking at protein interactions one-on-one, but over the time I realized that it is not possible to look at one-on-one protein interactions to understand functions. So something like this can be deconvoluted to something like this, a smaller network and a conglomeration of subnetworks. You can resolve this into structural details. There are 2 proteins, they interact with multiple partners. They may use the same interface. I hope people understand what is this interface. When 2 proteins interact, there is a contact site that is called as interface. And when you understand this, what you can do is that, for example, when 2 proteins interact through this interface, you can identify a region and make molecules that can disrupt this interaction. So if you identified some protein interaction which was important in, say, cancer, you can find inhibitors that are based on this interface, and hopefully that will inhibit the function of that protein. Sequence and structure carry the information for interaction. That is the design principle of my lab. And one look at it, you can see how 2 proteins can interact through multiple ways. Some of them through the same interface, that is the same contact site of one protein is used for multiple interaction. And that becomes very important in a situation like cancer and in the talk that I'm going to give. And I'm also particularly interested in something called a short linear sequence motif. Why? Because they have the capacity to encode functional interactions. They can be in the disordered regions of proteins, the ability to function independent of the tertiary structural context, and they also have a tendency to evolve convergently. So these short sequences are about 3 to 10 residues. You might think that they can be very promiscuous because 3 to 10 residues, how can you derive specificity from them? But majority of biological interactions are driven by such recognition, and they can be of the primary sequence type, they can have a secondary structural element, they can also have some tertiary structural element where this region covers most of the surface of the protein. But these are particularly not very valuable when you design inhibitors because any of these can be important. Something like this, if you can identify, it is very important because now you have understood, you have narrowed down the interface to something that is important. And when you make mutations, you can understand which is valuable for interaction. And you can design inhibitors. And something like this, a helix that has been used in the context of MDM2 to derive small molecules of inhibitors of P53. Most of you who know cancer will also know the protein called as P53. It's a tumor suppressor. So these are some of the definitions for SLIM. Don't worry about these long list of definitions, but SLIMs can be either modification sites like in phosphorylation. They can be ligand binding sites, which is what we will talk about today. There are zip codes that are recognized by cellular transport machineries. And viruses are very experts in using these SLIMs. And you can see in every pathway of viral entry, immune response, and transport, they use these kind of motifs to their advantage. And what happens if you have a SLIM like this? An in-frame mutation, which is enriched on one interface, or an in-frame mutation enriched on another interface, this can be important in disease 1, this can be important in disease 2. So these are some of the diseases that are known to occur because there are mutations in SLIM in some of the domains that proteins have and recognize these short motifs. This is an interesting case of SARS-CoV. Most of you would have heard about this Delta variant and the original variant and how it became more contagious and was spreading fast. This is a site. You can see that it is in a disordered region of the spike protein. All of the ones that are not contagious or didn't spread or infect human beings that badly carry this RS sequence here. SARS-CoV had this sequence, which is PRRARS. There is an RS, but it also introduced PRRA. Now, what happened is that this site is preferred for enzymes which actually cleave this sequence. And why is this important? This short sequence motif is important because a furin-like enzyme or a transmembrane protease, when it cleaves the spike protein at this site, is when it gets activated. So it's not enough for the virus to bind to the receptor. It also has to be cleaved and then activated to enter the cell surface. So when this proline was mutated to an arginine, one of the mutations that occurred in the Delta variant, this is known to be a better furin cleavage site, and the virus became very contagious and spread very fast because furin could act on this. And the most optimal site for spike, the furin site, is when the alanine is replaced by a lysine. This hasn't happened. This happened, and we had predicted this, that the current form of the COVID doesn't have the most optimal furin site, and it is really an issue that if it got mutated and became more preferred by furin, then you will have a problem. So this is for mutations and insertion. What happens in cancer is that something like a normal cell, this imagine to be a protein-protein interaction, you have one protein interacting with multiple proteins. Most often in cancer, what happens? The levels change. Now, if the levels change, which is now indicated in a bigger circle, what you didn't have previously, you might gain, or you might gain and get a new interaction, or whatever was previously present, you might weaken the interaction because now you gained a new interaction. So this is called as rewiring of the network, and this leads to all the hallmark cancer properties in cancer. So I'll quickly run through two examples, then I'll come to the main topic. So here is one of the proteins. Don't worry what this protein is. It's a oncoprotein and called as Gankerin. It also happens to be a proteasomal assembly chaperone. This protein binds to an ATPase, which is part of the proteasome pathway. And you see here the knob-like structure that is coming from this S6 ATPase is locked into the hole in Gankerin. Now, this is a short sequence motif, which is made of the sequence EEVD. So we identified this in the complex structure. You might ask, what is the application of identifying these films? What I can do now is that once I know this is the short linear sequence motif comprised of this sequence, I can go and find out in other proteins whether such a sequence is present. And if such a sequence is present, I can ask whether it is interacting with this Gankerin. And that's what we did. Padma in my lab did this and identified 7 interactions which carry the same sequence. When you mutate this, you abolish the interaction. We establish EVD is a conserved hotspot site for Gankerin interaction, and therefore any peptide or peptide derivative of the sequence can be a potential inhibitor of Gankerin function. We also validated the functional relevance of this interaction. But recently we also showed that the protein, one of the protein which interacts, if you mutated the residues EEVD to AAVA, and if you had a mutation in the Gankerin, which is converting the lysine to an alanine, you abolish the interaction. So there is a hotspot site on the client protein. There is a residue that is interacting with it, and we have shown both ways that this site is very important for interaction. We also showed that there is a small molecule inhibitor of Gankerin that we screened out from using structure, and that can also bind to Gankerin, and it is an intended target in the cells because if you knock out Gankerin or silence Gankerin, the effect of doxorubicin goes down. So we have a small molecule inhibitor, and we also have a short peptide. There were slightly overlapping binding sites, which I'm not going into the detail. Therefore, a chemist would be, the question to a chemist would be, can I now look at doxorubicin and convert it into a molecule that prefer Gankerin over other residues? Can I get a conjugate of the peptide and doxorubicin because they have an overlapping site? And can I get a synergistic inhibitor of Gankerin? Gankerin is a very important oncoprotein because it's overexpressed in many cancer cells. So this is what is the question: an overlapping druggable pocket. Can you find an interface peptide and a drug conjugate that might give you novel structural designs? On a second principle is another proteasomal chaperone. So this contains a domain called as PDZ, and PDZs bind to C-terminal residues. It doesn't matter what is the sequence of the entire protein is, whether it is homologous or not. if there are short sequences at the C-terminus, the PDZ domains bind. We screened libraries of peptides to identify interacting partners, and we fetched out these sequences that bind to PDZ. You can see one common factor that is similar in all these peptides. There is a hydrophobic residue at the C-terminus. And these are the proteins that interact with PSMD9. This we have proven that all these proteins interact, and they interact through the PDZ domain. They interact through the C-terminal residues. And we also, as I said, sequence and structure gives information on how interactions will happen. We have 3 different motifs. One, which are weak affinity. They tend to bind to the PDZ like this. This is what we hypothesize. Medium affinity. They tend to be anti-parallel to the beta sheet, but they don't make overwhelming contacts. But tight binders, they bind in an anti-parallel fashion like any other PDZ containing peptides, PDZ binding peptides. And what is interesting is that we identified this P-2 cysteine to be an important residue for interaction. No PDZ domain is known to bind to P-2 cysteine in such a preferred manner. And this interaction between PSMD9 and one of the protein is important for NF-kappa B signaling. But again, to a chemist, this would be important because that cysteine, we had a low affinity peptide, GRRF. There is a high affinity peptide, SCGF. By making permutation combination in understanding how it binds to PDZ, we converted this GRRF about 500 micromolar to 5 micromolar, and it required only one replacement, arginine to a C. And now we also have a dipeptide, GC, that can effectively bind to PDZ and inhibit this interaction. Now, is the cysteine really important? We did many mutations, many changes, and only one that affected is DTNB modification of the cysteine that reduces the binding capacity, but not the KD so much. So I don't know why it is important or how it is important. We are thinking about it. But if this were really important, this will be one unique PDZ where the cysteine is important. You can identify inhibitors that can block this interaction. Yesterday's talk was very interesting because you could have functional molecules that can target either to ER or the mitochondria by changing the functional groups. So here is a functional important residue, and can you make derivatives of small molecules that can inhibit this cysteine? So now I come to the topic that I had put forth for this talk, and this is, so one of the ways of looking at protein interactions is through this guesswork from the protein interactions, and if you have a knowledge on what are important for protein interaction, you could do this structural bioinformatics like I mentioned, and you can fetch the peptide, find common peptides, and see whether they are interacting, and you can go back to the protein. The other is to just pull down whatever is there interacting with the protein in the cell and ask what are the proteins that are binding to it by mass spectrometry. And this is the approach that we took for more comprehensive analysis. One important distinction, and depending upon what is the curiosity of the investigator, why this is important, because when you do mass spec, you can only pull down which are high affinity interaction. So you might miss what are low affinity, but definitely otherwise functionally important, like the short linear sequence motifs. But I'll give you a twist to the story as well. So we pulled out, we cleaned up, and then we found that there are 133 proteins which are interacting with PSMD9. So there is a question by everybody, what do you do with 133 proteins that are interacting with it? You can do some functional classifications. This is something that favorite for all students. They'll put these proteins in some cake pathway and they will get these. You have a spliceosome and ribosome, and the rest of the things, you don't know what to do with it. And if you looked at the networks, which are nice to look at, so you have these complex networks, again, what do you do with this? You don't know what experiments to carry out with this. But we did do one thing for the ribosomal proteins that are interacting with PSMD9. These are the ones that are pulled up by the IP and mass spectrometry. You can see when you knock out PSMD9 by CRISPR, the nuclear structure actually changes. So the importance of PSMD9 interaction in ribosomal protein becomes clear because it seems to be important in maintaining the nuclear architecture, because if you take out PSMD9, the homing is not happening and the structure alters. And that stabilizes P53. I won't go into the details because we don't have much time. And this, I wanted to just show to the students how you can validate something like this. Since it is PSMD9, it would interact with proteasomal subunits. PSMD9 binds to two proteins, PSMC3 and PSMC6, and that is a trimodular complex that gets enriched. And you can see it next interacts with the 19S subunits. You see this intensity that is coming down. So this is simply like what is the direct interaction, then what is the next indirect interaction. And intensity guides you to place the trimodular complex as the direct interacting partner. These are coming down with the PSMD9. Note that it doesn't interact with most of the 20S subunits. And you can show this in IP nicely. This is flat PSMD9, this is PSMC6 and PSMC3. These are flows through. Most of the PSMC3 and PSMC6 are captured by PSMD9. Now, what do you do with this clustered network? So how do you take out the noise within quotes, because you want to design experiments? This is what you do. We have experience with the short linear sequence motif. We asked how many of those limbs are enriched in this pull-down complex. We found about 56 proteins with the motif EXKK, which is highly surprising because this is something that is apparently missed in APMS studies. But we did find a motif EXKK that is enriched in multiple proteins. You can classify them. Remember earlier I showed the enriched cake pathways which showed spliceosome and ribosome. Now you can look at the molecular functions and it begins to make a little bit more sense because structural constituent of ribosome, it can be cadherin binding, it can be cell adhesion, it can be RNA binding. We have previous experience that we do have RNA binding HNR and PA1, which we fetched out by structural bioinformatic analysis. And if you take this EXKK and ask to which functional group they belong, translation elongation factor binding, unfolded protein binding, translation is highly enriched, unfolded protein binding is highly enriched. And here you see S14 is the ribosomal component, so it could imply translation. And HNR and PA1, which is an RNA binding, was also present in the structural bioinformatic approach. We took two clones which are overexpressing PSMD9. These are validation of those interactions that proteins which are enriched in the EXKK motif. I said structure guides you to do these analysis. You can see a structure of the complex of East NAS2 with the C-terminal of ATPase. You can see these arginine and glutamate residues becoming important at the interface. We made a double mutation of those residues, and what binds now at 200 nanomolar, the affinity is increased to 1.15 micromolar for two of the proteins that are interacting, thus validating end domain being important for this interaction. Now, any interaction or any such study should lead to the phenotype. And what are the phenotypes that we tested? We tested lipid biosynthesis because FASN is one of the interacting proteins that came up. It is also known in mouse studies, in a genetic study, that PSMD9 is important for lipid regulation. So this becomes our positive control. Unfolded protein binding, CNX, that is calnexin, is one of the interacting partners. Protein translation, there are several elongation factors that were there. In unfolding proteins, we took two, scalnexin and DNA-J, and asked the question, can we check the phenotypes now? In PSMD9 overexpressing cells, the lipid droplet assay, which is used by checking BODP, yesterday's talk again talked about a BODP which changes its fluorescence when it goes from hydrophilic to hydrophobic environment, and you can see that the lipid biogenesis lipid droplets are more in PSMD9 overexpressing cells. This is much more relevant when you knock out PSMD9 because the lipid biogenesis or the lipid droplets really go down, and that can be rescued by overexpressing PSMD9 in the cells. And for unfolded protein response, which is another phenotype that comes up in this analysis, you can see that we have checked two of the proteins that are known to be sensors of this unfolded protein response. And we see that in PSMD9 overexpressing cells, this goes down. This is actually encouraging because the other phenotype we saw was protein translation. And protein translation is enhanced in P9 clone 7 and P9 clone 10, indicating that when the unfolded protein response goes down or there is no stress due to this protein overexpression, and the protein translation go hand in hand. So what happens now is we have PSMD9 overexpression. It can lead to increased lipid biogenesis, and its effect is more in knockout, indicating that it is necessary for lipid biogenesis. Because the unfolded protein response is low, we believe that is probably due to transcriptional repression, allows protein translation to go forward. And maybe PSMD9 overexpression leads to increased anabolism. An unfolded protein response is also supported by the fact that there is increased association with 19S and less so with 20S. Therefore, the ubiquitin proteasome pathway is probably not affected because you need the catalytic core. So fatty acid synthesis, calnexin in the unfolded protein response, DNAJ also in the unfolded protein response. This is the network that PSMD9 is regulating, and that's what we have arrived at by looking at APMS data and introducing small linear sequence motif as a filter to pull out some of the proteins that are important. And these are the biological functions of PSMD9 validated in 2 different clones. We are pretty confident that this is physiologically relevant. We have shown a lot of studies in endogenous protein levels. This I'll skip for want of time, and I'll thank you. I think there is no need to summarize. I've been summarizing at every point. And I thank all my students who have done this work. And this is the lab. We don't know what we do, how we do, when things happen and don't think happen. And we are also funded by ACTREC, TMC, PDP, and ICMR. Thank you very much. I'll be happy to take questions.
So now the session is open for discussion.
So all these fantastic talk. So all these that you are looking at, the role of that PSMD9 and all. So in the context of cancer cell, because that's where you kind of started with. Okay. So when do you see? I mean, how does it connect to the phenotype or behavior of cancer cell?
Thank you. Because for lack of time, cut out all those data with MCF7, breast cancer cell line, where PSMD9 is overexpressed as compared to the MCF10A, naturally overexpressed. You don't have to overexpress like we did it in HEK293. We see these interactions preserved. We see the phenotype preserved. So whatever is seen in HEK293 is also seen in MCF7, and knockout removes these properties. So we believe that PSMD9 is giving advantage to MCF7 cells, breast cancer cells. And we also data from patients, which I skipped for want of time, where there was a network of RNA correlation. Maybe I can just. So this directly affects the proteasome structure. This isn't from the data from the normal tissue, and this is from the cancer. You can see that network is derailed, and the consequence of this is accumulation of 20S. And when you accumulate 20S, you either get a survival advantage because you're taking care of the unfolded proteins, and you have a problem with the ubiquitinated proteins. So we are trying to dissect what happens. And if you knock out PSMD9, the cells tend to grow slow. So the other interactions with UPR and protein translation, we see in MCF7 as well, and PSMD9 knockout changes this.
So I have a question. So you mentioned about the connecting the QI and the IPs and the the peptides with different affinities. So in your experience, what is the cutoff affinity where you will not see? I mean, what is the threshold? I mean,
for the peptide?
Yeah, like
between the peptide.
The pull down works, and then you are hitting that number.
So it's a good question. I don't know the cutoff, but most of the proteins that we pulled out from the structural bioinformatics approach, which are about 1 micromolar, you hardly see them, maybe with 1 peptide enrichment. Those proteins that have been pulled down by mass spec, they have an affinity in nanomolar. So the other ones are 1 micromolar because they are transient interactions. So my belief is that if you, by APMS, when you're pulling it down, you're pulling out strong interactions that are long-lived complexes. Whereas in the other kind where you've used motif as an you have fetched and the affinities are lower, we are seeing that it is more transient interactions. That's a hypothesis that we have.
So for those those stronger ones, you might want to do some other biophysical characterization.
It's going on. So these are all coupled with ITC.
Yeah, that's fine. That's what. Thank you.
Prasanna, fantastic talk. The thing you have just hinted on it, but really not gone into the details. So when you talk about the normal tissue versus the tumor tissue and the proteasome going wrong, but it eliciting the UPR still. Okay. So what happens with the unfolded proteins in that situation? Is the proteasome absolutely compromised, or is it overactive? What is going?
So the proteasomal activity as measured by peptide, fluorescent peptides, that doesn't change. But if you look at ubiquitinated proteins, it accumulates. Okay. So there is a problem in degrading that. And there are, and if you knock out, you accumulate more. So when you compare MCF10A with MCF7, you have more ubiquitinated proteins in MCF7 despite more proteasomal activity. And that is enhanced when you knock out PS79. So this is a complicated situation that there is some direct effect and there are some which are downstream that is complicating the process. And one of the questions that is always asked is that, okay, you pull down all these proteins, is it even relevant? You might have just pulled down because you overexpress the proteins. My argument is that what this says, you should consider HEK293 as a bucket where you put all proteins and then try to pull it down. Even if it is a cell, don't forget it is a cell. I had a soup and I put something and pulled out. But then what it says is that these are thermodynamically feasible. And the context will come. Maybe in one certain cell type, the context will be evident. In the other, it will.
Thank you.
I'm just curious about when you talk about slims, are you referring to only those short fragments as individual motifs, or those could be part of a bigger protein? Because the reason why I'm asking is when you look out for such kind of motifs in the entire sequences, how do you know that the same sequence is in the same structural context as a linear motif? How do you go about finding out that in a protein where whether it is in a similar context or a different context? That because I believe that that may change how they would interact with the proteins.
I agree. And it is not just in the peptide, it is in the context of the protein. And we have seen that if it is a C-terminal, is it generally disordered, but it is in the internal, like the TXKK motif, it can be present in the internal regions. There it takes up either a helix or a loop. We have found all kinds of structures. And so we don't know in the context of the protein-protein interaction, apart from this sequence, what else determines the interaction as well on that. How I look at this is that, so if you twist out all the way the protein interacts, there is diffusion, then there is a contact, and there's a random contact. They go around by feeling all the surfaces. Then they find their complementarity, charges, van der Waals, they stick. And that other secondary interaction, more proteins, reinforces this interaction. So if you looked at this way, then the slim in the context of making specific pools of protein interaction becomes significant. And if you fail to interact with that recognition sequence, even if it is of low affinity, you have a chance of inhibiting this and getting a success in the therapeutic context. So you really don't have to go for high affinity interaction. That's the thought process.
Thank you, ma'am, for this excellent talk. I would like to ask that whether there are multiple slims present in a protein or
you can have. So the same sequence.
Each one will have some specific interaction, or only one will
Yes, yes, they will have different interactions, and people have tweezed out some of the slims for particular domain, WW domain, PDZ domain, SRC domains on the same protein. So you can have multiple slims that are interacting. And this is also particularly important in proteins that function as scaffolds, where they assemble many proteins and they come together for, there is a trigger, they come together, they do the job and then dissociate. You will see that in that context.
Thank
you. Okay, if there is no more question, let us thank Professor Prasanna Venkatraman for her excellent lecture. Thank you, ma'am.
And thanks everyone for active participation. Small crowd is good.
Now Now I would request the session chair to kindly present a memento to Professor Prasanna ma'am.
So the next lecture is by Dr. Rijit Samal. He is from the Institute of Mathematical Sciences, Chennai. His research interests include complex networks, network biology, and system biology. But it is not there. It is. Where is lecture 2? It is not there in this. Okay, sorry. No, he is here, but this information is not in the sequence here. I don't see that. Okay, it doesn't matter. Okay, so the next lecture is by Professor Sangram Bagh. He is from Science Institute of Nuclear Physics, Calcutta. So the topic of his presentation is molecular engineering of living cells for building biocomputers and artificial neural network. With this, I request Professor Bagh to deliver his lecture. Professor Bagh, please. So your time is 25 minutes, and then there will be some discussion.
Good morning, everyone, and so thank you to the Arithmetic for this wonderful meeting, and I'm really honored to be here and try to present our work. So at SINP, my lab works in a field which is basically called the synthetic biology. The whole idea of the synthetic biology is we want to use actually the engineering principle and adapt it into the molecular biology regime. And our goal is to so that you can actually program a cell or engineer a cell or an organism in a similar way and engineer programs a device. So that is the bigger idea of the synthetic biology. But to go yeah. Yeah. So when you think about this engineering, you always looks this kind of the example. But I'm sure you never thought engineering like this. So the question, if I ask that, why at this technological era, when you can actually control the rover on the Mars from the Earth with almost a mathematical precision, why we cannot make a Hulk or the Pegasus? But the answer is very simple. For any of these things, you do have a very well-established engineering principles. You use these engineering principles to fabricate this and test it. And if there is a problem, you redesign your things and you develop that. However, we do not know what is the design principle of the life. And that's why we cannot make this kind of a thing. And that reason is definitely the complexity of the biological system. It is too much complex, so we cannot do that. So that's why today's genetic engineering and the biotechnology is basically mostly trial and error kind of the things, or we think about some qualitative idea and then try to engineer a system to get a different kind of the behavior. So the whole idea of the synthetic biology, that if we can just start to rethink, or in this way, that we want to design some of the biological function inside the cell or an organism, the same way engineered that design them. So that's why we can, if you think about hierarchical principles of computers, you start with some physical layers and then make several logic gates out of it, and then connect that logic gates to the modules, that modules to the computers network, and so on. So similar kind of hierarchy are also in the biology. You start with the protein and the genes, you connect to the biochemical reactions, those biochemically distinguished pathways, cells and tissues, and so on. However, the problem is from here to here, so how this evolutionary principle, how it connects from this scale to this scale, we have no perfect idea of that. Okay. So, for example, when I see that Chandrima and Oishi, they're talking among themselves, but when we have some eye contact, they just pretend that they're listening to me, right? But so from this, protein and genes, or this whatever the epigenetics or the mitochondria, we cannot say this kind of the behavior. But anyway, I'm just making the things, they're listening to me. But either way, we cannot actually say why I'm behaving like this, starting from this protein and genes here. So, but what if we don't know this principle, but we actually try to take this principle from here. For example, using the protein and genes, we make some biochemical reactions which will exactly behave like gates. So in this kind of the electronic gates, our input and outputs are the flows of the electron, either there is there or there is no there, or there's a threshold. But here we can think about that a biological gate where your inputs are some chemicals or the biomolecules, and your output is also some chemicals or biomolecules. So that means using this protein and genes, engineering them, we develop some biochemical reactions which use like a gate. But this principle, we do know that how these gates has to be connected to give a specific function. So our biochemical reaction, which works at the biological gates, you connect them to make a pathway, and we know what the function is going to give that. And we can go in this hierarchical way. So that's how, this is a starting point where we can actually start that to develop a new kind of function inside the cell, and which is coming exactly from this engineering point of view. Now, I just give an example, very old example in 2012, and I just want to make the case that why in the conventional biotechnology and the genetic engineering, you cannot make this kind of the system. So here, what you take, so different cancer cells have different microRNA signatures, but this is not a single microRNA. You need multiple microRNAs, and they have a kind of an expression as a signature, which is different than the other cells. So here, for example, the HeLa cell, this is a microRNA signature of the HeLa cell, which is different than any other cancer cells or the normal cells. So people actually develop, using the biological gates, something called a classifier circuit, exactly whatever the gates are connected together, and then put in the HeLa cell. And if this microRNA signature, which is going to match with this classifier circuit, then this circuit turns on, produce a protein, and kill this cell. But if we put that circuits in a normal cell or any other cancer cells, okay, there is no profile matching. So it is not going to kill that particular cell. So this kind of function, if you want to develop, you have to come through this kind of the engineering design, and that is how the synthetic biology is different than the conventional genetic engineering something. Now, just come to what we are trying to do at SINP. So we are working on several problems, and I'm going to talk a couple of problems that we have solved very recently. And one of the goal of our lab, or one of the questions we are asking, that if we can make bacteria artificially intelligent. So artificial intelligence is a very buzzword nowadays, and this artificial intelligence, most of the artificial intelligence what you find everywhere, that's based on something called artificial neural networks. And these artificial neural networks are coming from the neuron and our real neural network. we have a lot of molecular understanding of the neurons, but we do not know how these neurons are connected and making a lot of the function like our thinking, our imagination, love, hate, and so on and so on. But what we do know, if you take a neuron, either from a worm or a cat or a human, and you give some input in terms of the electrochemical way, x1, x2, and something like xn, and if you get some output in y1, y2, and yn, they always follow a similar kind of mathematical relation. And then when you just write down in the pen on the paper what is this kind of the mathematical equation, you start from this, you give x1, x2, something like that. The first, what they will do, they will combine linearly, mathematically linearly. So there is something called weight. So how they linearly combined, x1 w1 plus x2 w2 plus x2 w3, something like that. Now this function is now will go through a nonlinear function. Most of the cases is a nonlinear function, and you give an output. Now you connect this kind of artificial neuron together. What you get is called the artificial neural network. And this looks simple, but it is an amazing power. For example, most of us probably have our accounts in the Facebook. Now, if we take a photographs from this conference and just upload it there, and suddenly the Facebook will tell you, do you want to tag your friend this one? Do you want to tag me? Do you want to tag him? Do you want to tag her? So how Facebook is identifying, because in this Facebook server, they have this kind of artificial neural network, much bigger. They have a 60 to 70,000 nodes for facial recognition. So if you have a photo, so from your face, they're taking the pixel values and put as an x1, x2, x3 as a input. And now they will do all this mathematics here, and whatever the output is coming, it will not match with anything. So what they will do, they started with some W values, they just try to change the W values. Okay, it's not, there is a lot of algorithms and stuff that how we are changing the W values, but they are actually scanning the W values from minus infinity to the plus infinity. It's not true, but in that big range. And at a certain point, suddenly this output will match with your face, which we have already been uploaded in the Facebook profile picture when you are actually starting that. That's how it actually do that. So, and the problem is, so in all the artificial intelligence, mostly 95% of them, the people use in the computer, so in the software level, right? The problem is, if you have a robot, you bring a robot and want to take a decision, then you have to connect that robot to that kind of the server. But if you really want that robot, that everything is happening in a robot's head, then you cannot do through the software route. You have to go through basically in a hardware route. So okay, the idea, so you develop an artificial neural network in a chip where they will connect exactly like an artificial neuron. People actually develop a lot different kind of neuromorphic chip like this. People tried the photonics to do the ANN, and also people actually showed that even in the in vitro DNA computation, you can actually develop an ANN. Now, my lab is asking this question: can you develop the bacteria as an artificial neuron? And then in a culture of the bacteria, this bacteria will interact with each other like an artificial neural network, actually solve some of the computational problem. So we able to develop in the just end of the last year the first artificial neural network with genetically engineered cell. So this is the first engineered cell which can work as an artificial neural network, and it came as a cover article in Chemical Science, and it is also featured in Nature India. So to do that, so what we have done, first we take some of the computational function. So for example, two functions we have taken several functions, but I'm just in decoder and encoder. So decoder encoder is a very important function in any communication device. So if we communicate through a walkie-talkie, so whatever we are giving the signal is first encoded and then actually transferred. So you have a receiver, that encoded signal is decoded again, and then it is coming like that. So what I'm just trying to say, that decoder and encoder is very important for this kind of a communication device. So here we have a 2 to 4 decoder, a very simple decoder. So if you have a two information, X and Y, they can be arranged in 4 different ways, these two information. And for 4 different information, you have 4 decision lines. Okay, so it looks like that, and the electronic structure will look like that. So we don't want to go through the electronic design, but what we bring that we want to develop this as an ANN. So first we develop a set of rules that how from a truth table, if you want to map on artificial neural networks way, what would be the map? So here, basically, it is just nothing but something we written on the pen and paper, and there is the mathematics. So now idea is, can you translate that mathematics, that abstract mathematics, put inside the cell, in real living cell, where you design this molecular interaction in such a way that molecular interaction actually follow this kind of mathematics. If you can do that, and that is, we can develop that ANN. So for example, here we take, so this artificial bacterial neuron we name as the bactoneuron. This is just a name. So for here is one kind of this bactoneuron, for example, and it should give this kind of the behavior, and this is a mathematics. So this mathematics is translated in a molecular engineering point of view. So you have a lot of synthetic promoters. Synthetic promoters, they're not available in the nature. So basically you take the data box and then puts a lot of different sites for different transcription factors. Either it is activating or either it is actually repressing. And then whatever is coming out of this promoter, they can feed back to the other promoter and this and so on. So this is the first thing. So now when we actually have that, and your inputs is the chemicals in terms of like two small molecule chemicals, and your output is the fluorescence protein. So this is a busy slide. I'm not going to explain everything. What I'm trying to say, so first time when you actually do that experiment, whatever the input and output relation, it is not matching with the mathematics what you are asking. You feed that with the mathematics, and the W values, or whatever the weight, this is called weight values, they are not matching. They're exactly like the Facebook. Now you have to change the W. The way we are changing the W, we do start doing this molecular engineering. It's a lot of molecular engineering doing, and we are optimizing the W values in that way. So at the end, we find what is our final bacterial blue. I want to tell you. So here are the three different bactoneurons. So if you see the simulation, an experiment, simulation, experiment, simulation, and experiment. So both in all these cases, you see the simulation and experiment match very well. So now we have a library of bactoneurons. Now we want to mix and match, and then want to develop this kind of the function. So basically, when we are optimizing the weight, we are basically controlling the transcription, translation rates, DNA-protein interactions, the number of molecules inside the cells, and we have to bring in the appropriate parameter level. So now this is the result. So we made 4 different bactoneurons. You mix in a culture, and then this is the result. IPTGN, ATC for 4 different kind of information. You'll get here the fluorescence. So that is why you can see here diagonally there is 1, 1, 1, and everywhere is 0. Here diagonally is the fluorescence. Everywhere it is the black. So so this way we can able to make an 2 to 4 decoder. So similarly, we have made a lot of other functions, the multiplexer, demultiplexer, encoder, and so on. I'm not going to details, but this is the way we do that. And this is the encoder, that's experimental results of the encoder. This is the experimental results for the Feynman gate, the Friedkin gate, and so on. So that is the things, and we're very happy. Now, what we want to do, we want to expand the capacity of this ANN to do the more complex function. So one of the very long-standing challenge of the synthetic biology, can you make or engineer a bacteria so they can add? So can bacteria add numbers? And so for that, you have to develop then adder. So adder is a kind of the circuit or system which actually adds the binary numbers. And there's a similar way we actually in the decimal system. Only the difference is like, so you have a 0 plus 0, you can actually add. So there is a sum, and then there is a carry. The similar way in the decimal system, when it goes beyond 10, we have something in the carry, we put in the other. So you have a carry out, and there is the carry in. So this is things we try to develop this with the bacterial, this ANN structure, and we able to make the first full adder with bacteria. This is yet unpublished, but we are very excited. So similarly, we ask this question, can bacteria subtract numbers? And we develop, again, they use the difference and the borrow, the same principle we use in our decimal system. In the binary system also, they have a similar kind of system, and we're able to show that it can subtract binary numbers. So this is the subtractor. This is, again, unpublished results. So now, here we have a multiple cells, multiple engineered bacterial cells. They can do a lot of computational function. The question is, can you do this computation among different kind of the cells? So bacteria as well as the mammalian cells. So what we have done, we develop a system. It works like a Feynman gate, and basically you give some information to this bacteria, and this bacteria compute that information, and then that computed information is now transferred to the cancer cells, the HeLa cells, and then another level of the computation happens in the HeLa cell, and your output which come as the silencing of the two genes or two targeted genes inside this mammalian cell. So this is after published, this also highlighted in Nature India. So here you can see this is this pin-like structure, which is the engineered bacteria, engineered E. coli actually attacking the HeLa cell. And so if you see the results, so your input chemicals were applying on the bacteria. Okay, it's a co-culture system bacteria, but your output is manifested by the silencing of two inherent genes inside the HeLa cell. So this is a multicellular system between the bacteria and the mammalian cell. So another example I'd like to show from our lab. So maize is a very simple problem to solve. A child can solve this kind of maze. But if you're a wizard, this maze is not for you. You need the wizard maze, like Harry Potter. So Harry Potter is a 10 years or 11 years old boy. So any maze, either it's a wizard maze or it's this kind of the maze. So child can solve this kind of thing. The problem is, if you bring a computer or a robot and ask them to solve this kind of the maze, they cannot. We do not know how human brain actually solve this kind of the maze so fast, but computers cannot, because computers has to go through some algorithm. And now in 1971, this is the first maze solving computer made, and it looks like really, really very complicated. So what the question we asked that, okay, so at this moment we can develop a lot of different devices through this kind of the genetic circuits, okay, not just us, but many other people. So why not? We can now develop a kind of a computer which can solve this kind of the maze problems. And what we try to develop is a maze solving computer with the engineered bacteria which can solve simple maze. The simplest maze is the 2 by 2 mazes, and in this structure you can have 16 different maze problems, which have 13 have no solution and 3 have the solution. So now if you mathematically you want to put in the computer, the way you do, so you have a problem matrix. In this problem matrix, let's say if there is a open space, you call them as a 1, and if there is a closed space, you call them as a 0. So that's where you can actually develop a problem matrix. And in the solution matrix, if there is a path, you can say it's a 1. If there is no path, there is a 0. So when you have a problem and problem matrix as well as your solution matrix, now you have developed different kind of algorithm inside the computer, and computer can start solving. What we did, we just put it in a chemical space. So now we have 4 different chemicals, and we want to put in this. So if there is a chemical present inside the in the system, then it is 1. That is means it's an open space. If it is not, it is 0. And similarly, in the solution space, it is a 4 different fluorescence proteins. If there is a path, the bacteria will fluoresce. If it is no, then there is nothing there. So to solve this kind of system, we developed this kind of the electronic circuits, and this is the 6 different 4 input AND gates connected in this way. And this is the molecule, again, this is the molecular structure, what is the engineer, the way we actually engineer, do optimize, and so on, so on. And at the end, we have this 6 E. coli, so we mix in a culture, and that is your biocomputer. And I can show this is the results. This is the 16 problems. 13 have no solution. That is why you see it is black, and 3 have the solution. You can see there's the fluorescence coming there. So with that, we can actually say that now the bacteria can actually solve an abstract computational problem. And we're very happy to say that this actually highlighted in the MIT Technological Review, as well as the many other international science and technology media. And even Wikipedia put it as one of the notable achievements in biocomputer technology development. This might not be very great, but it is very exciting for us. Our students are excited that whatever we're trying to do, it is not only give some repercussion in the synthetic biology community, but probably in a bigger community, the other common people, because when it is coming featuring like that. So the question is why we are going to why do you need to do that? Why do you need to engineer a bacteria or any other cell to make the computation? The thing is simple, because you do not have a computer or a robot exactly at the micron scale. So when you think about the micro robots, they are always at the several centimeters. So why do you need several centimeters? Because you need sensors, you need decision-making circuits, you need actuators. At the very same time, you need energy. That's why you need a battery. And you cannot bring a battery in the 1 micron or 2 micron. Now, you think about the cell, either bacterial or even the mammalian cell, they're really in the micron level. Okay, you do not need a battery, you can give just a food from the outside. Now, if you can actually program them in such a way that they can do the human design computation, that means now you have a computers at the micron level. So now, the completely uncharted territory, there's a micron level computation, and there's a lot of applications you can think of starting from the medical and any other. And our lab's goal is to how to expand this kind of the computation with this. And our first achievement, I believe, that till now, last 20 years, synthetic biology is working on the electronic circuits way, and we showed that, okay, ANN is another computing system. So we can bring this ANN into this synthetic biology, and we can actually develop a lot of complex functions like the adder and subtractor, which cannot be done. This can be electronic circuit. It would be so complex to make that gene circuit. So with that, I'll almost finished my talk, but I have just 1 or 2 slides because we're at SINP. So what, as a whole SINP, as a biofraternity in this SINP, so where we are going, what is our future goal? So we are a basic science institute, but now we feel that we have a lot of offshoots of the research through we can actually want to develop a bio-entrepreneurship and the incubator at this. So the idea is to make a lot of startups and develop testing and validation of the new technologies which can come from our all biology research group. And we do have a very specific plan for next 3 to 5 years. We want to develop some of the cell models for different biotechnological and medical importance as a product. We want to develop some preclinical drug development pipeline and a lot of assays and protocols for biochemical markers for nutrition and the health. And definitely our goal is to facilitate and encourage the build startups and in-house faculty and the students. And I feel that's all, as I found yesterday, that all DAE units have a startup or this kind of the bioincubator. We are very excited. We don't have any experience. So we hope that DAE will support our initiative, and I'd like to talk all of you all the institutes who have the startups, because we need the guidance for that. And with that, I finish here. This is my group, and definitely my acknowledgement goes to the DAE for the most of the funding, Science Nuclear Physics, HBNI, and definitely some of the other government agencies from where we get the funding. And thank you very much for the attention.
So now the session is open for discussion. I think there will be many questions.
Yeah, thank you for the wonderful talk. I was just wondering, so the cells that you're using are E. coli most likely, right? So they would be moving in your culture. So is there any particular arrangement you fix them to, or?
No, so in these cases, there is no specific arrangement. So there's just a suspended culture. But what you say, so for so one of the goal, our next goal is basically, so at this moment we showed that I didn't talk about one, but this is a single layered thing we have shown ANN. But ANN would be the multi-layered. So now when we want to do the multi-layered, we want to do in a solid culture. So in that way, there is, but they are random. They will be random, but still that would be the fixed. So we don't have to arrange one by one like that. So it is a mixed culture.
One other question is that how are these weights adjusted actually in your
weights adjusted? So this is a molecule. Yeah. So first when we actually do the experiment and see that we know that what are the weight ranges to give that particular behavior. Let's say it is 20, for example, or 20, 25, 30. There's 3 weights, and now we are getting maybe 2, 5, and 6. Then so in equation you can see that we need a more sharper. So increase the weight means we have to give more sharper behavior. So to give that sharper behavior, we found that if we change the position of the transcription factors in this promoters and the numbers, then we can actually make this kind of this start. So that's why we're changing the weight one by one, find that, okay, it's increased a little bit, we do again. And sometimes we have to also change the relative amount of the proteins and the promoters inside the cell. So all the kinds of way. So it's manually we are actually changing the way.
So thank you for an excellent talk. I would like to ask whether, I was wondering whether these can be done for mammalian cells too, and whether it will make it more complex because of the splicing of it and introns, exons.
Yeah. So the people do the synthetic biology, the mammalian cells, and we have started. So first thing I have shown is that the HeLa cell. So there is a part of the computation is also done with the HeLa cell. So now we are going to the mammalian cells because so mammalian cell is complex in one way, but it is also easier in another way because mammalian cell has a lot of molecules through which you can actually activate or repress or something will actually diffuse to the other cells. So and that way we have a lot of weapons. So that is why we're doing a lot of things in the mammalian cell is much more easier. So that's for example, people actually developed in last 2 years that adder, full adder, because there is they engineered 32 different kind of the cells and there's a lot of communication things. But in the bacteria, we do not have a lot of communication channels. So that is why making that adder in the bacteria is the challenging. So yeah, so mammalian cell people are definitely doing. They have a lot of medical applications for the mammalian cells. They have a lot of things. We can discuss maybe the offline there.
Thank you. Thank you.
Wonderful presentation. It's really opened a new field for all of us. Okay. What I wanted to ask you was, is it possible to bring it down to single cell level instead of having, multiple cells?
Yeah. So we used to do. I mean, when I started my lab, so we used to do in the single cell. But the problem is that when you are increasing the circuits, so then it is difficult to do in the single cell. So that is why, I mean, when very complex kind of the thing, it is better to actually distribute it. So this is the idea of from the distributed computing. But yeah, the people are also trying in the single cell, but we do not know how in a single cell how big that can be done.
Because what kind of volumes are you using here, or for bacterial cells, or to have an idea? Because finally you want to take it down to nano level, right, for the micron level. But when you have so many bacteria, so that it no longer becomes just at a micron level. So what the volume or the areas, I just wanted to know you're dealing with here.
Yeah, so at this moment in the culture, but when you think about in an application, for example, you want to develop some system which you can inject in the blood and then go. So maybe few hundred cells. Okay, so here is not the area is not important, but the the single micron things are important. So that way we are thinking. We are not thinking anything in the nano way, but the way you are talking, maybe we can actually think in that way. That's definitely a new way to think. Thank you very much.
Thank you.
Yeah, actually, I was wondering, like, how would this bacteria behave, or the computation behave, if it's in the form of a quorum sensing, where cells in the peripheral stage, have a different kind of physiology than the cells which are like exposed to the different nutritional conditions.
Yeah, so I mean, definitely that is the important point. So how it is exactly working at that level. But the whole, whole point of our goal is to actually see a robust system which will work for several days. And when we actually get this kind of the average result, so we are happy. Now there is a possibility that there is a lot of huge amount of the variability among there. So the way we got, so we do the flow cytometry. If it says input and output, what is the, so you give an input, and when you do not give the input and want to see the output, if there is no overlap in terms of the variability, we believe that, okay, they are separated enough and we can actually use that. But there is many ways that to reduce this variability. And in those cases, probably we have to study something exactly in the peripheral way, how it is diffusing, and those kind of models we are not using. The reason is very simple. In engineering, the idea is everything has to be very simple. Whereas when you do the biophysics, so we want to do each and everything, because when I was doing my PhD in the synthetic biology, we used to, for simple mathematics, we used to write like 27, 30 differential equations all simultaneously. But the whole idea is to make it the engineering. So now we come to like 2 to 3 kind of the equation, like a transfer function. So definitely there is a gap in biophysics and things. But yes, but that's why probably we want to collaborate with many people, even the optics, and how what I was saying is very, very important to actually go there. Thank you.
Yeah, I have one question. This when we talk of engineering this bacteria, have you thought of doing this for the probiotics? Because they are already good bacteria and they're fighting many diseases. Suppose I can understand to start with E. coli is the best organism, but is it possible to?
Yeah. So there is a lot of companies are also there who developing a lot of kind of engineered probiotics. For example, in synthetic biology, so in general in the medicine you have a diagnosis, and then doctors prescribe some medicine and something. So people are developed a lot of different kind of the cells, okay, including my PhD. So where you take a disease states, maybe in the metabolic state, maybe some other chemicals. So if this kind of the things, like people develop engineered probiotics, this is not in the clinical stage though, but if there is a salmonella is attacking your body, or so they give some sending some molecules. Your engineered probiotics actually sense that molecules. When they sense these molecules, they turned on and produce a specific peptides which will only kill that salmonella, but not any won't touch others. So this is an autonomous kind of it's called program therapeutics. So it's an autonomous kind of the therapeutics, and people are also going into that. Yeah, but yeah, we can think about a lot of applications because we are thinking it from the cancer gene therapy point of view. But we need a little more students and someone who can work
Just
on that.
one more quick question.
Yeah.
Is these attributes that we are engineering bacteria, they are transferable by any means transferable to other organisms?
Other?
Suppose you have molecularly engineered bacteria we have developed.
Mm-hmm.
The attributes that we have imprinted in this bacteria, are these attributes transferable? I mean, by any means can they transfer to another hostile organism? Suppose my engineered bacteria transfers this capability to a hostile bacteria, then that bacteria becomes super villainous.
So that is why this engineering, most of this engineering we do in the plasmid. We do not change the. In one case, we just change the chromosome because we put 2 transcription factors there, and just transcription factors. So that is why it's a plasmid. So when you do the experiment, and there is not any pathogenic protein and this. But okay, when we design this E. coli which invade the cancer cell, so there is a pathogenic protein because that is your cancer cell actually identified. But those are BLS2 biosafety level 2.
Fine.
So yeah.
Thank you.
Thank you.
Dr. Sangram, excellent talk, and I'm realizing that my neural network has slowed down, so I'm trying to catch up with the talk still. So some part of the question is already answered. So the question that I still remain is dependence on spectra for detection as an output detection. So can that be, can there be innate signatures which can be the detection methods? Then, because there may be some specific advantage to that. So something like innate metabolite or Raman spectra or that kind of thing. So do you have any thought that?
Yeah, I mean that is
what is the need? That also I'm thinking still.
Yeah, so we do this fluorescence protein because this is very easy to do. I mean, the first thing. So whenever we have a circuit, we actually characterize everything with the fluorescence protein. Now, for example, when you show that the cancer cell, when there is also a part of the computation is happening, their output is not fluorescence protein because this is the output is the silencing of the genes, and that we can actually do in the qPCR and Western blot. But what you are saying in terms of the Raman spectra and something, So that is important because I know that synthetic biologists who are working on bioprocess engineering. So when you have a reactor and you have a couple of proteins as a product, then those kind of the things, the only thing you can, so the people have a lot of IR sensor and Raman sensors, those kind of things. So that is very important when you actually scale up or at a different level. So yes, people are using the Raman spectra. I do know people are using IR spectra and all kind of, but not.
They are already in use.
It is because these are very standard techniques for the already for the reactor. You are just using that one as an output of your system.
Yeah, it's an excellent talk.
Thank
I
you,
have
sir.
just one question. In neural, you were talking about that neural network is used for this, you're solving the problem in the optimization. And you were talking also that it is just like a pattern search method you are following to exactly minimizing or optimizing the thing. Is it really pattern search? Because that will be huge number of possibilities. Then it will take a lot of time. Or you are rejecting certain routes and find it out. How do you select that?
Okay, so definitely when you talk about the artificial neural network in the software, so doing this kind of the pattern search, so they have the algorithm to change the W's, and that's a lots of different methods for that, and there is a lot of positive and negative.
Yeah, I was asking about that. How do you decide that?
Oh, so here we are. So now we are not using this. So at this moment, what we are doing. So let's say we are doing this ANN in the pen and paper. So if you do that ANN in the pen and paper, then you can also change the W's by yourselves, right?
Yeah.
At this moment, our goal is to make this hardware. So at this moment, the way we changing the W is by manually by engineering, because we know that. So at the very beginning, we found that just from a mathematical equation, what could be the range of the W's or the weights and bias. And now when we actually make something, we got some wrong weights and the bias. Now we start optimizing it manually by doing the molecular engineering. But our next goal is that what you're saying. So we want to develop in such a way that it could be autonomous. So in that case, at this moment, what we thought about is using, because if there's a cell-cell communication, so there is a diffusion of the quorum sensing molecules, and that diffusion constant, and because this cell maybe sit here, this cell, I mean, if there are two cells are there, if they may sit here, so based on this distance and the diffusion, the weight values could be different. So this is a kind of a parallel things. That is we thought about.
So decision making will be done that parallel.
Parallel.
Good.
Yes.
Excellent work.
Thanks.
People have many queries that can be discussed during the tea time, lunch time, and he's here for 2 more days. But I have a question. Just it will be slightly different, but I thought let me see these biomolecules, or let us say secondary metabolites. Their biosynthetic pathways are well known. But suppose if I want to make a molecule, a small molecule, whose biosynthetic, it's not a biologically made molecule. So whether this system of coding and all these things can help. Let us say I want to synthesize, make benzene. For example, let us say benzene is not made by any biological pathway, but I want to make it. Whether this system can help.
So not this system. In the synthetic biology, there is another part which is called synthetic metabolic engineering. So there is like you said, so this is called the retrosynthesis. So they develop a lot of different pathways to make a different of the molecules which is not produced by this organism. So there are two ways. One is, let's say there is a malarial drug called artemisinin, and this artemisinin is very costly because you have to extract this from the thing. But the one group actually developed that if they take the pathway from that plant and put it in the yeast and E. coli, so that make the artemisinin, and now this artemisinin price actually came down. So this is one way. Another one where the people are working, which is not also present in any other organism, or I mean not plant or something, but they want to see, let's say, how if I want to make a carbon to carbon double bond, so what are the enzymes and how this pathway, because it is not just a pathway, what important is that the kinetic matching has to be there. I mean, if we just put some genes and it is not going to make that. So we have the kinetic matching. So there are groups which are actually doing that, so that is why I mean, you are a chemist, so I am very afraid to talk about that. The synthetic biologists sometimes think, so okay, we want to replace the main chemistry and everything will be on the 37 degrees, so that it is a green chemistry. But yes, people are trying on that. Thank you for that question, sir.
Thank you very much for excellent talk.
Now I request the session chair to kindly present a memento to Professor Sangram. Thank you.
It's an honor, sir.
The next talk is by Dr. Arijit Samal. Dr. Samal is from the Institute of Mathematical Sciences, Chennai. His research interests include complex networks, network biology, and system biology. Dr. Samal has been awarded with FEBS short-term fellowships in 2009, CNRS MPG Joint Program in System Biology Fellow from 2010 to 2012, Ramanujan Fellowship from 2016 to 2021. His scientific recognition include Simon's Associate of the International Center for Theoretical Physics, that was from 2015 to 2021, Head of Max Planck Partner Group in Mathematical Biology 2017 to 2023, and DAD Research Ambassador 2018 to 2025. So now I request Professor Samal to deliver his talk. The title of his talk is A Phytochemical Atlas to Harness Traditional Indian Knowledge. Professor Samal, please.
Thank you very much for the kind introduction, Professor Ghosh. Let me start by thanking HBNI for giving us the opportunity to come here and experience this beautiful place. And I would like to really express my gratitude and thanks to Professor Majumdar and all the organizers of this meeting for their both warm and generous hospitality. I think we don't want to be inside but outside most of the time, but I think we have to be somehow. The title of my talk is A Phytochemical Atlas to Harness Traditional Indian Knowledge. So before I start, I just wanted to say that the work I'm going to present is sort of a partial area that we work on. So since I started my group in IMSC about 8 years back, over the years we have developed working primarily on 3 directions. One, we work on compiling, curating, and exploring chemical spaces where chemicals could be of importance for drug discovery, systems toxicology, or human exposome. And actually the piece of work that I'm going to present today will essentially fall in this drug discovery space. But we also work on exposomics, and if I have time, I would briefly mention what we have done in that area. You may be expecting me to do modeling in mathematics or whatever. So we do develop tools and particular tools to analyze complex networks, in particular biological networks. In that area, we are both developing tools to analyze network, and one of the areas that we are focusing on is developing edge-centric measures by taking concepts from geometry and topology. But we also continue to work on modeling sort of gene regulatory networks. And a third area which we have been working on is also systems modeling of metabolism in microbes and protein secretion system. I hope to come back and talk about that. So these are sort of projects we lead. And we, of course, have expertise in aiding experimental groups. So in this space, we basically play an enabling role with experimental biology groups where they have data, high throughput data or high dimensional data, which we help them analyze in the network context and come up with hypotheses. So I just wanted to mention that I would like to stick to time. So there are two posters by my students today. So the talk I'm going to present today, the extensive results and all are available in poster 25. So welcome to ask questions. Maybe we can save some time at the end of my talk. And also in the second space, you may want to see the poster 26 where we are discussing modeling gene regulatory networks. So with that overview of what we do in my group in IMSC, so I'd like to just slowly dive into the area that I'm going to present today. So essentially over last 5 to 6 years, we have been developing this area, which I broadly call as compilation, curation, and you can think computational exploration of chemical spaces. And broadly, we are focusing on two types of chemical spaces: the natural product spaces, in where you may be interested in chemicals which are potentially beneficial to humankind. And here I would present our work on the phytochemical space. I won't have time, but we have also developed spaces for medicinal fungi. And we have also worked extensively on chemicals which are environmental chemicals, which could be potentially harmful to the humankind. So I won't have time here, but I would have loved to present that as well. So with that short introduction to the sort of the research that's going on in my lab and the broad area which I will present today, let's dive into what I'm going to say. I don't think this audience needs justification for working on natural products, but as you know, natural products continue to be of immense interest to scientists working in the field of drug discovery. And mapping and exploration of these natural product spaces is important for the discovery of new therapeutic molecules. So you have a chemical universe, you want to look at the biologically relevant chemical space, and within that, the natural product space. And as you know, phytochemicals or plant secondary metabolites, they play a very important role in the case of plants in helping them adapt to their ecological niche. Moving on, plant extracts, as we know, contain phytochemicals, and they have been used to treat human ailments across different civilizations for centuries. Moreover, many of our present-day drugs, which are approved by US FDA, they happen to be initially isolated from plants. Morphine, the first drug which was, got FDA approval, FDA in its earlier avatar, or artemisinin or paclitaxel. Also, I should mention that we have estimated what fraction of small molecule drugs approved by US FDA to date are based on natural products, and it's about 40% as we estimate today. So moving on, various systems of traditional medicine, in particular both Indian and Chinese, use medicinal plants in their formulations. And there's a lot of interest here as well, I understand, in sort of tapping this knowledge. So what I would argue is that when we started thinking about this, and it was just for fun actually, we didn't really wanted to work on this area, we were doing for fun over the weekends. So we realized that the information on traditional Indian medicine, I'm not going to particularly talk about any Ayush system, and in particular the phytochemical space, the information is largely buried in books which are not in electronic nature. And I felt that this non-digital nature limits its effective use towards modern drug discovery. We also recognized at that time, when we started thinking about it in 2016, that if you look at particularly the phytochemicals, in contrast to Indian herbs, there were much more digital resources and data available to be tapped for traditional Chinese medicine. And for example, there was TCMID, TCMMesh, which compiled a lot of the work. And with that in mind, we felt that a large-scale database, which compiles the information in particular about phytochemicals, could actually facilitate computational approach towards traditional knowledge-centric and natural product-based drug discovery. So with that in mind, we started by fun, but we created Impact, which was actually the first version, which was published in 2018. To the best of our knowledge, this is the largest phytochemical atlas in silico for Indian medicinal plants. So after we published in 2018, what I will talk today briefly is what we have achieved between 2018 and 22, when we have just recently updated the Impact in a big way. So I'll essentially focus on the expansion and the enhancements that we have achieved in the last 4 years. So currently, if you look at Impact 2.0, it compiles more than 4,000 Indian medicinal plants, almost 18,000 phytochemicals, and more than 1,000 therapeutic uses. So the new version was released. I mean, its impact is available through any of these links. It's released on 17 June 2022, and the full version of the manuscript is available on bioarchive. So before I move on to the enhancements that we have achieved on Impact 2.0, let's discuss what we had achieved in Impact 1.0 in 2018. So this is a brief summary of all the compilation and the exploration of the spaces that we achieved. So what we had in Impact is a compilation of information on more than 1700 Indian medicinal plants. And if any of these plants could produce any of these phytochemicals, it was 9596 phytochemicals we had. We also had information not on the phytochemical associations, but therapeutic uses, and some information on if these plants are used in traditional Indian medicinal formulations. So I don't have time to go into the detail, but after compiling this sort of the data set, and you need to realize that we don't keep in our database any chemical for which there is no structure. So that is, it needs to be computable. So we did 3 different analyses to sort of make a case that it is a very interesting space to look for potentially for drug discovery and wellness research. First, we showed that there is a large drug-like space, and this drug-like space, if you look at, almost 60% of it had no similarity with then FDA-approved drugs. Further, there is only a very minor overlap between the phytochemicals for Indian medicinal plants and Chinese medicinal plants, and the overlap was only 25%. And also, if you look at the drug-like space, it is only a minor overlap. Thirdly, I don't have time again to go into details of this analysis, but we have looked at in detail the distribution of various physicochemical properties for various sort of natural product spaces and compared them with commercial synthetic and semi-synthetic compounds. So one of the analysis which was inspired by the work of Stuart Schreiber, which comes out is that the natural products really are very different in the distribution of shape and stereochemical complexity, 2 size independent measures. And what Schreiber had shown is that for his sort of a benchmark natural product library, they were able to show that natural products are less likely to be promiscuous bonders of human target proteins. And based on this analysis, we also were able to suggest that the natural products or phytochemicals in our resource are likely to be also very specific binders to their potential targets if you're designing drug around it. So that was Impact 1.0, which we published in 2018. It was really done with fun, for fun. We really didn't think we'll work on it, but pleasantly we realized the significance and utility once we published the work. I had never built a database or did any of this kind of work at that moment. So what we have done over the last 4 years is sort of work diligently in expanding and enhancing this database. And this, I would say, it's a resource, and I'll try to quickly walk you through what are the different expansions and enhancements that we have achieved in the last 4 years. First, there has been a very extensive digitization effort. So we need to realize that the information on phytochemicals and the quality information is not really in published literature, but actually in books, that is not in PubMed. So we have digitized more than 100 books in the last 4 years. We have also revisited more than 7,000 published research articles and several existing resources to compile this database. Further, we have achieved increased coverage. We have more than doubled the Indian medicinal plants and almost doubled the phytochemicals in our database. And if you look at the associations, it is more than 5-fold. Lastly, an important advancement that we achieved is that if you're working on plants or plant extracts, the phytochemical content varies between different parts of the plant, from leaf to root to bark. So what we did is that we actually now compile the information at the level of parts. This required a lot of manual curation effort. We have actually standardized to 27 different part IDs, and the data is available at the part level, both phytochemicals and therapeutic uses now. So I would like to just acknowledge that again, this paper is available. You can read everything, and importantly, my PhD student was submitted and is waiting for his thesis to be defended so that he can join his position in US. Vivek actually led this work, and Karthik, who is now in Dresden, he's doing a PhD there, and Ajay, who is here, who will be presenting the work. So they have really contributed heavily to this work at the expansion part. So let's look at what we have achieved. So one thing I wanted to stress is that it may seem mundane, but you actually need non-redundant information. So we have a non-redundant list of 4,010 Indian medicinal plants. The way we went about doing this, it was really manual, is that we compiled 7,000 synonymous names of medicinal plants used in various traditional medicine. We mapped them to their accepted scientific names, then merged them and have a non-redundant database, and then look for their phytochemicals in various sources. So what we then achieve is that not only the information is non-redundant, but then we are able to give better taxonomic classification used in traditional Indian systems of medicine, synonymous names, and actually we also compile information if they are endangered based on the IUCN Red List. So there are various results that you have. Majority of these plants, of course, have been used in Ayurveda, but many are actually have also been used in Siddha. And you can see some of the plants actually require conservation efforts on our side as well. So having moved on the sort of coverage of the medicinal plant information, you can actually, we went about improving the information at the level of phytochemical content. Here I want to emphasize that in this round, we realized because once we published 1.0, we became aware of many other books. People just would contact us and tell us about this rich resource and all. So we actually improved the database by digitizing 70 specialized books for phytochemicals. And if you're wondering how many pages there are roughly 40,000 pages across these 70 books. And I will show you a picture of the 145 books we have done, which is about 82,000 pages that we have digitized. So this information was important because we wanted the information at the level of plant part, and that we gathered from these books. And also we had to revisit every data in Impact 1.0 because we wanted the part level information. So what we achieved is a non-redundant stereo-aware library. So that's another thing we have. We developed a workflow. I don't have a time to go into to really sort out the stereochemistry of the phytochemicals that we have in our database. So in principle, we have this 17967 phytochemicals, but they are stereo aware. So there is a workflow we have developed to actually resolve that as well computationally to the best possible. Finally, what is enhanced more than the expansion is that we have enhanced annotation of the phytochemicals. In particular, we have the molecular scaffold information now, which will be very useful. And also we predict using a tool the biosynthetic pathways which could potentially produce this phytochemicals. So if you are wondering what are these books, these are some of the books that we have digitized. So this is the Wealth of India, this is the Compendium of Indian Medicinal Plants, and there are many more. So the supplementary material of the paper and bioarchive also gives all the books that we have digitized, more than 140 if I'm not wrong. So lastly, so we have the plant phytochemical information, but we have focused also on the therapeutic uses. I should have mentioned that we, as you know, if you go to do a natural language processing kind of search, use a plant and cancer, most likely every plant will be linked to cancer because somebody puts a grant like that, okay? But so we purposefully avoid such information being put into our database. So we only take therapeutic use information from standard books recommended by Ayush Ministry. Don't take it from such data. And furthermore, this information is not standardized. Again, there's a problem of the same symptom could be used in a synonymous way. So we map it to a non-redundant list of therapeutic uses and map it to essentially the identifiers which are used in Western medicine. So we have in this round not only MESH but ICD-11, UMLS, and disease ontology. We will be soon releasing the SNOMED mapping as well. So we have standardized these therapeutic uses from various sources to 1095, but the number of associations are almost 90,000. And finally, what we do in a pilot scale, because we don't want to reinvent the wheel, is to also capture some information on the use of these plants in traditional medicinal formulations. So as you may be aware, there is a large effort by CSIR known as the Traditional Knowledge Digital Library, where they've been digitizing these traditional medicinal formulations. They claim to have digitized more than 2.5 lakh. Only 1250 is openly accessible. It's not good for a small team like me to reinvent the wheel. So we only link if a plant in our database is used in the traditional medicinal formulations 1250, which are open. I should mention that we have also digitized the few pharmacopoeias, which we will be soon releasing also and putting it up in our database. So again, the information is at the level of parts now. So that's essentially the data. I can't give you all the details or the different levels of information we have, but Impact has a very friendly user interface. You're welcome to go here or impact.com or impact.in. So the way to access the information is three. You can either browse by plant, phytochemical, or therapeutic use, or you can do basic searches and you can do advanced searches. For example, now you're able to do advanced searches based on your molecule structure versus our molecules. If something is similar, you can actually extract out. So in some sense, the advanced search option is kind of a simplified cheminformatics platform to extract information relevant for you from the resource. And at the end of, I mean, you can play with your phones if you are interested. You get all the 2D, 3D, and other information that we have compiled. So till now, I've only mentioned the data that we have compiled. But as you know, I'm in Institute of Mathematical Sciences. I'm not supposed to compile data, so I'm supposed to use the data. So let's talk about what we learned by using the data. So the first thing is that, is there anything interesting there? And I am not an expert. I'm not even a novice, but my student is an expert on, Vivek is an expert on cheminformatics. So he actually was a master's in cheminformatics, so we are able to do it. So we essentially decided that there are many sort of cheminformatic measures or filters which people use in the initial drug discovery pipeline to filter out molecules of interest, small molecules of interest. So we use these 6 different measures which we coded. And I said, let's take a wisdom of a crowd's approach and ask which of the chemicals pass all of these 6 measures. And we found that about 1,335 pass all these 6 measures in our updated database. And what is very interesting is that if you look at these, 11 are already FDA approved drugs, that's good. But in the expanded database, almost 90% of the drug-like phytochemicals have no significant structural similarity to any FDA-approved drug. So I will mention the scaffold part in a couple of slides, but you may say, okay, they are different, but are they really molecular scaffolds? So we have done a very detailed analysis of scaffolds and now it's available. So if you look at the overlap of the scaffolds itself at different levels between the IMPACT phytochemical library and the drug-like phytochemical, the US FDA-approved drugs, the overlap is very little. So you actually have very different chemical scaffolds to sort of explore in this library that we have created. So that's the first result that we show, that there is a untapped potentially drug-like space in IMPACT. Second is again the same question appears, every reviewer wants the question that how much is the overlap with traditional Chinese medicine? So I mean, we could do best with whatever is the best resource available. So the open resource which is available, TCM Mesh, compiles about 10,000 phytochemicals for almost 6,235 Chinese herbs. We took that data, we did the analysis of potentially drug-like phytochemicals there, and we did basically a direct comparison as well. Essentially what we find is that the overlap is a minor overlap. There is unique scaffolds and unique sort of phytochemicals to explore in our phytochemicals associated with Indian medicinal plants. So lastly, we have also done in detail this time a scaffold identification and annotation. So I have no time to go into the details, but we have taken the definition from this and we have scaffolds at 3 different levels. Here you see you don't have the identity of the nodes, that is the atoms. Here you have the atoms, here you also have the bonds. So we can take at that. And one of the ways to ask is that when people are interested in this field is that how diverse is your library? And a very simple measure, and there are many more detailed measures in our paper which we have done, is to ask how many unique scaffolds are there per molecule in your library. And in that range, we have compared from 100 million compounds in PubChem to all approved drugs and many other natural product libraries. We find that IMPACT is actually in this comparison of about 10 libraries, 3rd highest in terms of scaffold diversity. I don't have time to give more details, but for example, these are about 200 plus scaffolds which are highly represented in phytochemicals in IMPACT database. And you can actually browse our database and essentially take information out in that fashion. I will end in 5 minutes for sure. So, so where are we? Okay, so I just wanted to give you a glimpse of the results that we've obtained. So moving forward, if you think about it, where are we now is that we have the information in IMPACT at the level of plant part now. What are the phytochemical content of that plant part? You also have quite good information on if the plant part was used for some therapy. Okay, but the idea would be to sort of link the phytochemical content to the target proteins and explain the therapeutic effect. Okay, that's the long-term goal. So, and in a as a computational lab, our long term is to provide a state-of-the-art platform, which we believe already IMPACT is, but improve over time, slowly and surely, and make it a one-stop place for academics and industry working in traditional Indian knowledge and natural product-based drug discovery and wellness research. It is also important to recognize that digital resources like IMPACT would also serve an important role in turning a largely experience-based enterprise into an evidence-based science, which there's a hope for in the Ayush system. So I'll just end with 3 slides. I just mentioned that we have extensively talked about IMPACT, but we have a very interesting resource on medicinal fungi. We have done similar analysis here. And this is, I think, in the world, the only resource on medicinal fungi secondary metabolites. And this is another natural product space which people could tap, which we have created. And finally, from the beneficial to the potentially harmful, we have been working a lot on characterizing the human exposome. So many of you may not have heard about the term exposome. Exposome is essentially the environmental complement of genome, and there is a lot of interest in developing the human exposome project. So human exposure to environmental chemicals has been realized to be an important contributor of disease burden, and we want to understand that in our daily life. And interestingly, there are recent perspectives which call for both network science and computational approaches towards linking the external chemical component with the health effects. And we have actually been working a lot in that field. And I just want to highlight that we have at least 5 different resources. So we have on endocrine disrupting chemicals, neurotoxicants, human milk contaminants, and so on. And this work was led essentially by Janani, who's completed recently her PhD, and B.S. Karthikeyan, who was there. So I'll just, last bit of advertisement is that, I mean, this work also has got attention. So we have this resource called Deduct, which I think is one of the most extensive we have built. This was built and we have curated 792 environmental chemicals which could be potentially endocrine disrupting with dosage values and so on. This work required us to go through 22,000 papers, full text. So, and what is pleasing is that, of course, India is still building its REACH regulation and so on, but the French National Agency for Food, Environmental and Occupational Health and Safety is drafting a policy for France of what chemicals should be should be banned. And last year they released their public report, which is about 85 pages in English. And essentially by looking at 27 different worldwide initiatives, they chose Deduct to be there, and they took our chemicals and are prioritizing based on that. So this is, I think, one of the maybe, I mean, it's very nice to see that a blue sky research in India is affecting Western policy. With that, I just want to also mention that why I mentioned this is because many of you are really interested in not only phytochemicals, what they combine is potential toxicity. So these tools that we have developed actually can be very useful in building models to predict toxicity of natural products, and I don't think there are very good predictors at this moment. So we would be exploring there as well. So essentially, with that, I'd like to thank the people. The students who contributed are listed. I mean, Vivek is really out, he's waiting his defense, and these were some other people who contributed. These are sort of my collaborators elsewhere in other projects. And I want to also thank the public funders who have funded our work. And with lastly, I just want to mention that I want to thank you all for the attention, and I welcome questions if there is any time and suggestions, and I will be happy to collaborate. Or I hope people use the resource. That's all. Thank you very much.
Now the session is open for discussion.
Hello, it's a nice talk. So I want to know actually these active ingredients of different plants are different from region to region. So as a natural product chemist, if I want to extract the active ingredient from some plant, so can I get some information that from which part I should take so that I can get more amount of that compound?
Yeah, very good question. So, I mean, there is also the issue of cultivars, another thing. The only answer I'm giving is that I'm just giving you the phytochemical content at the level of part, but I'm not giving the amount, right? That is also what you would like to know.
Yeah.
So I can only say that we are in the process of a smaller scale, about 600 plant essential oil data, which we are coming out very soon. Ajay is leading it. So we will be releasing that. So what is the essential, I mean, the key to that resource is that we only selected papers where the samples were taken from Indian subcontinent, because that's what you're worried about. If it's grown in Iran, it may have a different particle. So we'll coming with that resource very soon. But it's only for about 600 plants because data is scarce there.
Thank you.
Hello. So what I want to ask that suppose I want to search a chemical moiety with specific stereo details. Now I want to search your database and want to know which of the phytochemicals contain that chemical moiety with that particular stereo details. Will you give it?
So you can take a sort of a smile and search it. It will give the top 10 compounds similar to it, and then you can download those compounds and look at the results stereochemistry. The inches that we provide are resolved for stereochemistry. It will give you the top 10 compounds. We don't want to give extensive computation because this is not running on a very good server currently. So we allow only top 10 to be thrown out. But we can do such things. But I should mention that all the codes that Vivek wrote for this work, we have put in my lab GitHub page. So you're welcome to take them and do it on your computer.
It's a nice talk, Majid. And I'm just wondering, so when you have taken a lot of data from already digitized platform, so have you used like any natural language processing things?
So in the first round we did, but the problem is that natural language processing can at best give you, some sentence which suggests that, but what is the evidence? I mean, when you dig deep in the full text, the evidence is not that solid. So if you look at these monographs which have been published by people like Asima Chatterjee or really stalwarts, that is really trustworthy. So I don't want to put in noise in the resource. It's better to have little information but trustworthy information rather than have a lot of information with crappy information. Okay. So we went for that sort of a balance, if I say so.
Okay, I have another question. So let's say some other people developing this kind of things for the Chinese medicine or something, for example. So is there any way that those things can be integrated with your things so that we have a much bigger...
Very good question. I think, see, this version 2.0 is released in CC by NC. So it's free for academic use, but not for commercial. So I don't think I can happily tell you that the open source Chinese tools like TCM Mesh are nowhere now close to our work. You can look at them. They don't even have a proper web interface. So we are way ahead. But I am told that there are several companies in US who have developed their phytochemical databases, which you can subscribe to for $10,000 a year. So I don't know if we match them. I have not looked at any of such resources myself. Yeah, thanks, Sangram. Any other questions? Thank you very much for your attention.
So if there is no more question, let us thank the speaker for the exciting stuff.
Now I would request the session chair to kindly present our memento to Professor Arijit. Thank you.
Now the last speaker of this session is Professor Chandrima Das. Professor Das is from Saha Institute of Nuclear Physics. Her research interests includes epigenetic regulation of chromatin structure. The title of her talk is Chromatin Readers as Regulators of Cancer Epigenome and Their Therapeutic Potential. So Professor Chandrima Das, you will deliver your lecture. It is for 25 minutes.
Okay. Good morning, everyone. At the onset, I would like to thank everyone for being in this meeting and the crosstalk which we are having for the last one day. It is fantastic. Really thank the organizers for inviting me, as well as HBNI for giving me the podium to share our research interest. RRCAT is a really very nice campus. It is very beautiful, and we are having a very nice time. So our laboratory focus is on dealing with a particular type of family of proteins which are called chromatin readers. And here, okay, so if we consider that genomic information is compacted in the tiny confines of the nucleus, and the genome is actually into different organization levels. Some are compact regions of chromatin or heterochromatin, and some are looser region or euchromatin region. And so in these regions, the transcription programs are very different. So this transcription programs or the gene expression programs are guided by small modifications that happen into nucleosome. So what is a nucleosome? It is the core histones wrapped around by the DNA, and the histone tails which protrude out from this nucleosome organization get extensively modified. So these modifications are dynamic in nature. There are enzymes which are called writers, which puts in these modifications, and the erasers removes these modifications. As I mentioned, the marks are very dynamic, and our interest is on chromatin reader class of proteins who actually physically recognize these histone post-translational modifications and interpret their functions. So if you consider that this is a N-terminal tail of histones, you can see a plethora of different modifications, and these are some of the very well-known chromatin binding modules or reader domains that you can see here. One of our interest is on plant homeodomain kind of proteins, which is basically a zinc coordination complex forming protein, which has C3H, C4 type organization. And this is a protein which has very diverse kind of function, but histone modification, particularly methylation recognition, is one of its important role. The other class of protein is the bromodomain transcription factors, which harbor this antiparallel alpha helix with their loop regions. These loop regions are the sites which recognizes this acetylation marks, and these important recognitions are also translated in functional outcome in the cellular context. So reader function is evolving. They have roles in the context of autoimmune diseases, inflammatory response. We and several others are interested in its role in investigating the context of cancer. It has been shown to have important role in circadian rhythm regulation and aging as such, as well as stress responses. So varieties of stresses, these readers play very different roles. So what do we actually do in our laboratory with reference to the chromatin reader biology? So we are trying to understand the role of these epigenetic regulator proteins in the context of infection, metabolism, and cancer, understanding the crosstalk between these events, like how an infection-borne cancer and these reader proteins can regulate the functions in that context. And definitely one important trust area is the metabolic regulations in cancer. So with this, I will tell you very briefly 3 aspects that we are interested and would then focus on the story today. So as I mentioned, our laboratory is interested in metabolic regulations. So we know that when the glucose, which is the basic, the energy source in the body, if you alter these energy levels, like low glucose to short-term high glucose exposure to a prolonged high glucose exposure, very different biochemical reactions or metabolic programs are initiated in the cells, and there is a very nice coordination that happens within the cells to take care of these glucose fluxes. So in the context of cancer, it is also known that people who are diabetic have very high risk of developing liver or pancreatic cancers. So we are working on these regulators or these epigenetic readers and trying to shift the balance and such that the homeostatic of glucose can be seen. Another area that I was mentioning is the infection backdrop. So in the infection context, people are designing several different strategies to target the viruses. We are thinking in terms of metabolic regulations. So viruses as such really prefer the sweet genes. What I mean by that, they need extensive energy to harness to sustain in the host cells and proliferate and basically use the system, and then they can survive and amplify there. So they actually, in multiple cases, have been shown to regulate the carbohydrate metabolic genes, and very recently we have observed that gluconeogenic pathway is one mechanism which viruses actually hijack, and then they can use these chromatin readers as baits, or can kind of use them to program the epigenetic landscape, and then use these glucose metabolic programs to their favor and proliferate in the cells. Another just short glimpse that I would give is on the context of drug resistance. In therapeutic perspective, we are extensively working on the aspect of drug resistance and how these epigenetic regulators work. So one of the well-known mechanisms that most of you know is the action of the efflux pumps. So when the drugs are there by the cells, there are the efflux pumps which flush them out, and that's how you find that these drugs become insensitive to the acting cells. So in the cancer drug resistance perspective, we find we are trying to work out on the mechanism to sensitize these resistant cells, and we find in the therapeutic perspective, if we actually combine the epitherapy, then the situations can be salvaged to some extent. So like this is one example where the case in the doxorubicin treatment, which is, we know, a very standardized drug regime, and the cancer, particularly triple negative breast cancer patients. So what has happened is there is constant effort to titrate the dose of the drug down, so that now the off-target effects and non-specific effects can be reduced. And the patients also do not have the extreme negative effects of chemotherapeutic regimen. But what is seen that if you really titrate the drug down, you have very different mechanisms operating in the cells. So now the drug, instead of becoming a chemotherapeutic drug, is actually causing the reverse mechanism or promoting some of the cancer promoting gene expressions. So in this kind of situations, we find that the DOX treatment, along with a combination of epigenetic regulator, can bring the balance again, restore the balance and bring the function of the drug back. And now you can have the more kind of situation salvaged in that case. So now for today's talk, I'll just shift the gear a little bit. Instead of the triple negative breast cancer, where we have really been working for a while, we have been recently involved in the cellular fate commitment and how these chromatin readers can actually program the epigenome. So in this context, we're working on one of the very important proteins, zinc finger mint type 8 or ZMYND8, which is a chromatin reader. Why? Because it harbors these chromatin binding modules, plant homeodomain, bromodomain, PWWP motif, and the end terms, and it can actually recognize the different mechanisms, different chromatin, different genes in the chromatin context, and have role plays. So consider a normal epithelial cells. So here we know the E-cadherin or cadherin gene expressions are heightened, which is very important for keeping the cells in the normal homeostatic level maintained. So here this protein, in association with other epigenetic recognition functions of it, will go to these cadherin genes and effectively express the epithelial phenotype. On the other hand, if you take a mesenchymal breast cancer cells, this particular protein can actually is involved in regulating EMT, MDR, and cancer stem cell like genes, and that's how it can program. So this is what we have seen earlier. So overall, if we summarize the information, we understand that these chromatin reader molecules substantially can tilt the balance from a proliferative fate to a differentiated fate in a cell. So we see that in certain cases, the terminal differentiation markers, as well as there are certain expression of these, and the pluripotency proliferation markers goes down when the D8 levels are heightened, whereas in a low D8 expression context, the pluripotency and proliferation markers all get activated. So with this backdrop, we entered into neuronal differentiation mechanism, and this particular understanding was because we also work on neuroblastoma models. So considering the all-trans retinoic acid, which is a very important therapeutic agent to target neuroblastoma, can we have readers play a role in differentiation therapy and can transform the cells from the proliferative stage to a differentiated state. So this is a simple cartoon, master's level cartoon, that we know that neuronal stem cells give rise to the progenitor cells, either neuronal or glial, which can form the neurons, or the glial progenitors form astrocytes or oligodendrocytes. So very interestingly, it is observed that in the stage of differentiation, different epigenetic modifications are actually altering. All about the DNA methylation marks like 5-methyl cytosine or hemimethylated cytosine, and then acetylation and methylation modification of the histones, they are all altered with the stages of this differentiation. Interestingly, our also attention was there on the non-coding RNA pools. So you see the proliferative microRNAs are actually decreased, and then the ones that forms as mature neurons, those microRNAs are increased. Informations on long non-coding RNAs, however, very limiting in this particular context, and how the epigenetic readers regulate the expression of these long non-coding RNAs. So our interest on that arises because a few years back, we did a transcriptomics analysis after knocking down the ZMIND8 protein, and we observed that there are a lot of all-trans retinoic acid regulatable genes, indicating there is possibly a direct connection between the differentiation programs that it is relaying in the cells. So this is neuroblastoma cells. When we have the ATRA treatment, it forms the neurons. And what we observed that out of the several differentiation promoting genes, as you can see here, one of the genes caught our attention, that is MAPT or tau. This tau is the gene and MAPT is a protein. The expression was hugely downregulated upon this D8 knockdown situation in an ATRA-treated condition. So let us look that in the neuronal stem cell perspective, the D8 expression was found to be heightened in the neurons as compared to stem cells or in the astrocytic lineage. So now what we did is we targeted the D8 in the neuronal stem cell perspective, and we find that upon BFGF withdrawal, which is leading the neuronal lineage commitment, we find that there is a huge reduction in the expression of the tau protein when you target D8. So when D8 is not there in the system, the tau protein which was supposed to be expressed is not expressing anymore. And this was actually initially, as I mentioned, the expression levels showed that in the RNA level as well. So the question is, what is the mechanism and how D8 is regulating it? So this epigenetic regulator, if targeted in the context, and we did RNA sequencing experiment, we observed a huge number of differentiation promoting genes which were differentially regulated in by this epigenetic regulator. We subsequently looked at the genes that were actually regulated, the tau gene that was in our mind. So we found that the MAPT gene, the expression of it was most heightened in the brain. So now what is going on is the mechanism. So one simple mechanism could be promoter mediated regulation. It's a transcription factor, goes to the promoter and binds to there, brings the polymerase machinery and does the differential expression program. But there could be other mechanisms as well, because when we looked at the tau gene, there were several exons, as you can see, more than 17 exons with introns in between, and they were different, up to 24 junction processing that happens, and these 17 exons can be arranged in different ways. How many transcripts are formed? These many number of transcripts are formed from this MAPT gene, as we can see, out of which 5 of them basically are potentially non-coding transcripts. They do not code for any protein. So now, when we looked at the MAPT gene very carefully, we observed that within the gene, there were certain start site-like sequences and epigenetic marks, which actually was mimicking an initiation program from the transcription perspective. So we looked at this, we designed specific primers, which was to the promoter and this introgene regulatory region. Observe that D8 physically gets recruited this IGRR sites or introgene regulatory regions, and there it brings about certain changes in the classical transcription activation signatures like K4 methylation, K27 acetylation, etc. So now when it goes there into this MAPT gene, what does it do? It basically represses the transcription program. Shown here are the two important nucleosome remodeling deacetylase complex counterparts, that is CHD4 and MTA1. They form complex with D8. So what is happening here is D8 is physically getting recruited within the gene, bringing in a transcription repressor complex and shutting down the spurious transcription events that could have happened from within the gene. So now, if you look at the exon-specific gene expression by the MAPT genes, we find that there was transcription events that were very much tightly regulated in a D8 knockout background. So when you have gene-specific primers for exon 3, exon 4, exon 5 like that, and you find here there is this IGRR sequence that we are thinking about, and you have primers generated after this exon 4, and you find the heightened expression of these transcripts in absence of D8. So this indicates that new events are actually happening. So interestingly, we have to confirm that this is actually non-coding transcription events that we are talking about, because so we had generated antibodies for this tau protein in the N term, C term, or in the central region, the exon 5, and we observed that no protein coding is actually happening when you target ZMY-ND8 or knockout D8 lines that we generated by CRISPR-Cas9. There was absolutely no expression of this particular protein that is happening. So this is a non-coding transcript. We went ahead and we observed that this kind of internal initiation events that we are talking about is not only restricted to MAPT. There are several other genes listed here like TUB3, DRD2, SCN2A, lamins, everywhere. This mechanism does happen. So it's a very generalized mechanism of stopping the spurious transcription events so that the actual parental transcription can actually proceed. So since we did a total RNA-seq, we could understand that around 10% of the long non-coding RNAs pool were directly regulated by ZMY-ND8. And when we looked at the GPEX brain samples of the humans, we find that the correlation exists between the D8 expression and MAPT full length. But the correlation graph with the ZMY-ND8 and this MAPT213 is really poor, indicating the regulation is the positive regulation that you can find is with a full length transcript, but not with this 213 isoform, which is a non-coding isoform. The experiments were a little tricky because, as I mentioned, there are so 18 isoforms of MAPT. How do we design primers which specifically target this MAPT213 and not touch the others? So we had to do extensive sequence alignment and find out certain regions, like in 213, there are certain intronic regions which in the matured MAPT transcript are spliced out. But in the 213 non-coding RNA, they are retained. So we took those and used them for understanding what is going on there. So final experiment that we had to do to convince the reviewers anyway, that to clone this MAPT213 transcript and express in the cells, and this anti-regulation that I'm talking about is that if you see here, the MAPT213 the transcript level when overexpressed in the cells, the MAPT mature tau transcription is down-regulated, and when you complement back this full-length D8, there is again restoration of the full-length transcript and the reduction in the 213 transcript. This is happening in the transcription level, and we also looked at the tau protein matured transcript, and we can see the similar results as I mentioned. So there's an antagonistic regulation between this long non-coding RNA and the matured MAPT213. And to make things easier, this is the cartoon representation, as you can see. So this is a dual mechanism, how this chromatin reader is regulating the expression of MAPT. The standalone mechanism, which was already known and reported from our group, was the promoter-mediated regulation. Herein, we report that there is a new mechanism of IGRR-mediated regulation, where these repressor complexes are coming into play, and the repression of this particular spurious event can lead to neuronal cell lineage formation. And when you do not have this complex in the system assembled, because D8 is not there, then the cells can actually form this long non-coding RNA, which is actually inhibiting the parental transcription event. So there's a switch, dual mode of regulation, which D8 is doing along with the other very important neuronal repressor complexes in the cells. Okay, before I end my talk, I just would like to give you a very short glimpse of the kind of cancer research work which is ongoing in SINP to further increase the dynamics or interaction with us and the HBNI different institutes. So as three important aspects that we are focusing is early detection of cancer through multiple approaches. And in this particular detection aspect, definitely metavolumic profiling, which is one of the very important thing from the urine, serum, or blood samples. We're looking at the total metabolites by mass spectrometry and understanding that how this cancer detection can be affected. We are doing flux analysis. So we have this extracellular flux analyzers, and we measure the extracellular acidification rate or oxygen consumption rate. And then we can also conclude about how the the situation, the cancer, how bad it is. So we are doing artificial intelligence. You have heard a little bit from Shongram's talk today, as well as there are other groups also who are using AI as a tool for easier detection or early detection in the cancer context. Viscoelastic property measurements as well as nanotechnology are two other aspects which we are also utilizing for understanding the early detection of the cancer in the cellular perspective. Okay, so the second aspect on this is the simulation or characterization of the cancer. So here, if you see, this is a glioma model. So we have made this neurosphere cultures, which is simulating glioma, and we are looking at the mitochondrial energetics or the alteration in the mitochondrial architecture that is found in this context. We are also looking at the neuroblastoma perspective and how the intercellular junctions are playing important role in this perspective. We are also making spheroid cultures like liver cancer model. We have established the hepatic spheroid cultures, and there also we are looking at this powerhouse of mitochondria and how their architectural changes in happening in the hepatocellular carcinoma. In the breast cancer model, again, we have sphere cultures established as a 3D model, and you can see the proliferation versus the other properties like the hypoxic and nutrient-deprived core have certain marker expression, and the metabolic regulation, which I'm hinting at, is going on in a nice way to understand the tumor as a 3D model and understand the biology that is happening there. And finally, the highly relevant target identification. In that, we're using several important biophysical tools and also the electron paramagnetic resonance as well as the NMR as important tools for the detection of these important drug targets. We are using the potential anti-cancer drug targets for the DNA-G quadruplex measurements. We are also looking, this Shongram has mentioned, how we are designing synthetic biology tools to target the the cancer cells selectively. Okay, so we are also advancing the RA-based targeting strategies with conjugation of nanoparticles and trying to see if more efficacious delivery of this nanoparticle mediated RA is possible. And as I talked a little bit about the epitherapy, we have several of these epigenetic readers, the UBR7 and CBX4, and we are looking at the different hallmarks of cancer and how these epitherapeutic targets can be used basically to target and challenge the cancerous situations. So thank you. These are my lab members, and these are my members who have migrated, my students. These are my collaborators, my project funding agency, definitely funding support from Department of Atomic Energy, SINP, and others. I would like to take your question, and thank you so much for patient hearing.
Yeah, the session is open for discussion. Maybe.
It's a very nice talk. So when you treat with these retinoic acid, and then the cells differentiate from the neuroblastoma cells, do they continue to survive or do they die after they differentiate?
Very nice question. So it's very time dependent event. So anyways, RA treatment needs to be supplemented on a regular basis. And so we have different experiments that we design on the early differentiation program or late differentiation program. And where a very long time after subjecting it to the higher threshold doses, the cancer cells do die. So if you really put it a very high threshold dose. But what we are doing is we are trying to understand the molecular biology in terms of the day-wise programming changes that actually happen in the cell.
So the reason why I asked is that a strategy that one could use in cases where these are generally non-differentiated.
Yes.
And the other example is the AML where they give the arsenic tetroxide.
Absolutely. So yeah, so I believe these like RA treatment is even for the terminally differentiated, terminally diagnosed patients like the neuroblastoma patients or the glioblastoma patients. RA treatment is normally used still for them. So here we are trying to make it more efficacious in the way how we can improve the deliveries and all as a side measure, but we're also trying to understand the biology.
Thank you.
Is there any other question? Okay, Professor Dasi, I just wanted to know, means, what is the fundamental difference between this pluripotent cell and stem cell? Or
So,
this may be a very vague question from my.
no. Embryonic stem cells have the potential to form all the lineages of the body. So what I was talking about is neuronal stem cells, is a kind of a type of a stem cell which forms specific lineages. So either neuronal lineage or glial lineage, because they can give rise to committed cells. So it's a lineage commitment programs, whereas the embryonic stem cells as such can potentially form any lineage of the body.
So, if there is no more question, let us thank the speaker for the excellent lecture. And also, I would like to thank again all the 4 speakers of this session for their very exciting lectures. Okay, although we are running a little bit late in time. Okay, so again, I thank all the lecturers. Thank you.
Now I would request the session chair to kindly present a memento to Professor Chandrama Dasman. Now also I would request Professor Prasanna Venkataraman to kindly present a memento to the session chair, Dr. Sunil Ghosh. Thank you, ma'am. Thank you, sir. So now let us take a short tea break and we all will gather here back at 12:15 for the next session. Welcome back, everybody. Now, let us move towards the session 5 of day 2. And it will be my pleasure to invite Professor D.K. Maiti onto the stage to kindly chair this session. So, also, I would like to introduce him first. So, Professor D.K. Maiti is Associate Dean of. Oh. Professor D.K. Maiti is Associate Dean of HBNI. He is a gold medalist from Jadavpur University. He joined 33rd Batch Training School of BARC in 1990. He received his PhD degree in 1995 from Jadavpur University. He did his postdoctoral research at University of Utah, USA during 1998 to 2000. His broad research area is theoretical and computational chemistry. He has published more than 125 papers in peer-reviewed journals. He has guided around 7 PhD students, and he is also a recipient of Distinguished Faculty Award from HBNI. He is a Fellow of National Science Academy, India, and member of Academy Council of Mumbai University. I welcome you, sir. And I hand over the mic to you for the next session.
Thank you for your kind introduction. And this session has 4 lectures again. So first lecture is by Professor Subhasish Chattopadhyay from NISHAR. May I now request Professor? Yeah. Yeah. Professor Subhasish Chattopadhyay is a associate professor at School of Biological Sciences, NISER. His research interests include cellular immunology, cell-mediated immunity, inflammation, immune regulation, cancer immunity, infection immunity. He has been awarded with Sudev Bhushan Ghosh Young Scientist Award from the Zoological Society, Kolkata, India, and Rapid Grant for Young Investigator Award from DBT. He is editorial member of Journal of Immunology Research, guest associate editor in Frontiers in Immunology. He is review editor at Frontiers in Immunology, Frontiers in Cellular and Infection Microbiology. He is invited reviewer for Viral Immunology, Immunology, and Immunogenetics Insights. Okay. He has more than 49 peer-reviewed publications. He has supervised 3 PhD theses and has 4 ongoing PhDs. He is a live member of Society of Biological Chemists, Indian Society of Cell Biology, Indian Biological Society, Indian Immunology Society, and Indian Science Congress Association. With this, I request Dr. Subhasish Chattopadhyay. And your talk will be for 25 minutes. Then we'll have discussion after your talk.
Thank you, Professor Maiti. So, very good morning to all of you. And it's a great pleasure to be here in RRCAT. This is the second time for me. And really exciting environment, especially the ecosystem. And I really appreciate the organizer, convener, organizing committee, HBNI, all my senior brothers and sisters, and all the stakeholders for making a fruitful and successful meeting here. And so we'll be talking about my presentation. I will try to make a very generalized presentation, and I will try to go with time. So what I understood that the current day, the biology, it is expanding just like our universe. We all know the basic understanding about universe that after Big Bang, universe is expanding. So as for the science and especially for biology, what I wouldn't say that I'm a faculty of biology, rather I'm a student of biology. I am also learning and expanding my knowledge. So we believe in interdisciplinary work, where I believe no one is perfect, nothing is absolute, because in a context of relativity or relative context, we are all learner. So this is the main gate of Neisser. It's not working. So you can see the securities are there. Even in RR-cut, security probably is little stricter or tougher than Neisser. But security system is there, and our homeland security is like that. And when we talk about our immune system, it's just like homeland security, a kind of guard, where the outsider should have a kind of gate pass, or a kind of insider should have their own ID card. And no one is perfect. Still, we get kind of problem where there is a mismatch of the security guard, or including the invaders or outsiders. We get autoimmune diseases, same side goal. Sometimes our faculty is also, I remember in our old campus, a same faculty was asked and argued for 3 times. Also, he had the ID card. So now about biology or life sciences. This is our artistic presentations we had in Neisser. We have many students. We made these things. Actually, I was also a participant. This is from my hand. I don't like to say I or my. So we worked together, and this is around 7-8 years ago in Neisser campus. We made a kind of art and exhibition and competitions for biology. And you can see that it's so diverse. We don't have to explain what is what, but we understand it's just like a universe where it has a directional scattering. We are scattered, but we have a direction, and we need to find within this scattering the focus. So this is, as I mentioned, this is works like an autoimmune kind of, for autoimmunity, cancer, and infectious disease works like a shield. And these are the basic understanding of our organ systems, where it together contributes for a fruitful protections. But again, no one is perfect. Nothing is absolute. We get problem, and that's why the problems with altered immune systems can start infections, tumor, cancer, inflammation, and other pathological diseases. This is about basic understanding about immune cells, both adaptive immunity and innate immunity. And we all know that these T cells and B cells are the adaptive immune player. They are like commandos. Yesterday, Professor Majumdar, the convener, mentioned many things, and I like the word surgical strike. And for surgical strike, you need a precision. And these adaptive immunity cells, T cells and B lymphocytes, they have the precisions. And the other cells are pretty important. I would say that adaptive immunity is more luxurious rather than the innate immunity, where innate immunity is contributing for a kind of general guidance. But when there is a requirement of precisions, we need the commandos, and that are the T cells and B cells. And this is about typical cell-mediated immunity. What we call about final outcome is by T cells, where with antigen presentation, you can see that a kind of cartoon with signal 1 and signal 2 with MSC and TCR. The T cells get activated, and it can leads to inflammation or immune responses, or it can also go to immune suppression or regulation. And we are working on cell-mediated immunity and cellular immunology, which has implication in disease biology. These are the representative protocol or system where we work with in vitro, in vivo, ex vivo, cell culture, microscopies, and many other things in animal model as well as human. So immune suppression has a positive and negative impact. Sometimes it is beneficial. We have to have an open mind. Immune suppression is not always a bad one. Sometimes it is good, sometimes it is bad. Nowadays we call as immune regulation. It is good for autoimmune diseases where we get same side goal. Think such a way like where a kind of war-like situation is happening and commandos firing are coming to the civilians. It's just like autoimmune diseases. Whereas for infectious diseases and mostly for cancer, they work like escape mechanism. they work like terrorists. And you should know that the war between the terrorism as well as through a kind of open war is different. Terrorist follows an escape mechanisms. So cancerous cells do the same thing, because cancerous antigens are not immunogenic, and that's why they can escape, and we need to think for the next things. This is one of the editorial where we have talked about in health and diseases how cell-mediated immunity can work, most precisely different aspects of diseases, including cancer, infections, and autoimmune diseases. So this was the Treg. Now we call as regulatory T cells. Initially it was known as suppressor T cells. We worked on that. And if we can manipulate, because in cancer, finally we get solid, I mean, there are different type of cancers, scenarios are different, but the common paradigm for solid tumor progression is that we can see at the later stage after metastasis, a profound immune suppression. So we need immune protections and immune activation. So regulatory T cells is detrimental for cancer, and it facilitates tumor progressions. Now it's a suppressor. So the goal is how to suppress the suppressor, how to regulate those bad regulators. So this is the manipulation, and this was a kind of old things, but still it is relevant that when we propose that how our anti-cancer CTLs are getting inhibited by the regulatory T cells, where they generate a tumorous microenvironment which is immunosuppressive with different immunosuppressive cytokines, IL-10, transforming growth factor beta. So I told how to suppress the suppressor, how to regulate the regulator. We proposed and shown in human patients with melanoma that if we can promote a kind of protective environment with TH or type 1 helper responses, we can think for a protection, because TH1 conditioning can inhibit the regulator. They are the suppressor of suppressor. So you can get a protective responses. So our current goal, as I mentioned, as kindly Professor Maiti mentioned, we are working on infection immunity. I was trained with parasite immunology, but currently I'm working with viral immunology, and in collaborations, as I mentioned, universe is expanding. So biology, so as immunology. So we are working with chikungunya virus infections. We are working with experimental immune suppression in cancer environment, with also with immunosuppressive drugs, and then with T cells and macrophage. So they are the 3 PhD students who has contributed for our lab so far. And this is the presentation where the last year Nobel laureates got the Nobel Prize with TRP channels, David Julius and Andrem, who worked on different other channels. So we will partly mention about these things. Transient receptor potential channels is basically originally discovered as, this is also a collaborative work with Dr. Chandan Goswami. I call him with fun, he is my good colleague and friend, Mr. TRP in India. It's a ion channels, a non-selective cationic ion channels, which is known for thermosensation, but nowadays it has shown for different physiological role, and we look for T cell responses and macrophage responses. This is the work we have shown that if we inhibit this V1 channel, TRP-V1 transient receptor granular channel, it can suppress T cell activation. You can see that CD69 T cell activation marker is down-regulated with the pharmacological inhibitor as well as the inflammatory cytokines. This is the paper, and this is the cartoon where we proposed in 5th G that from naive T cells to activated to effector T cells, we need TRP channels, especially the V1 and V4. So it has importance for channelopathies and different type of inflammatory responses. Now coming to the textbook knowledge of innate immunity, as I mentioned, commanders are good. They are just like authority. For example, our authorities are here, but we need a group of people who can support us. So innate immunity support the adaptive immunity. But the question is that whether a commandos knows how to do a traffic signal, the basic things for a traffic police, the answer is yes, commandos supposed to know everything. Similar things we have asked, the innate immune receptor, that is the toll-like receptor we work, it is well known to act present in innate immune cells, whether this innate immune receptor is present in adaptive immunity on T cells. We have shown with that this is the paper where we shown this is a viral peptide, vaccinia virus peptide, A46 domain or A46 strain. We found that TLR4 inhibitor, this bioactive peptide, this is not a pharmacological inhibitor, by the way. It is reported to inhibit especially the TLR4 signaling in different diseases and innate immunity. This is the first time we have shown that it is also important for T cell responses. So that means the commandos also express the characteristics of a innate immune responses, like the common things, and that is pretty much important. So we have shown that with this inhibition, T cell activation is down. And these are the work. I will just quickly go. We have checked the TLR4 expression, and we found that with activation it is down. It is reported earlier, and with the inhibitor VP it is coming back. And for activations, we have found that CD25 and 69, these are the activation molecules, these are also down. And functionally for AICD, activation induced cell death, which is a part of extension of T cell activation, which is also regulated by this TLR4 inhibitor, toll-like receptor inhibitor, Viper. And the cytokines are also down. You can see the signature cytokines, IL-2, interferon gamma, and TNF are down. So these are the findings that we have shown, that importance and existence or functional expression of TLR4 in T cells. Now, chikungunya, we all know, I'm not going to the details. In India, we also face this epidemic, and there are different cells involved, and it has been shown that synovial macrophages and monocytes are one of the important immune cells where they can reside and replicate. And macrophage is the universal host cells for all these things, just like India. Macrophage is just like India, where all the outsiders can come and inhibit, there is no problem, and then India will shape by their own. So we have looked for the CMI responses, and this is the paper where we have shown first time that in macrophage, when we infect chikungunya, it induces apoptosis, and we got pain to prove that things that macrophage, there is no apoptosis through chikungunya. This is the first paper, and we have shown the importance of heat shock protein 90 using the heat shock protein 90 inhibitor, that it is regulating infection, inflammation, and other things, apoptosis also. So I'm not going to the details. We have shown caspase and structural and non-structural protein of chikungunya, and also the inflammatory cytokines. When we inhibit the HSP90, the chaperone, it is down-regulating infection as well as inflammation, and this is the things. And after that extension of that things, we have shown the involvement of those MAP kinases, that P38 and JNK are involved for this inflammation. And we found that P38 and JNK both are activated, whereas ERK is not. And with the inhibitors, we found that P38 and JNK inhibition down regulates infection and inflammation. We have also extended to T cell responses. And this is the cartoon we recently published from Taposh, that there, and I'm not showing the things. We have shown that physically it interacts, the NSP to the non-structural proteins is interacting with the P38 MAP kinases by IP as well as molecular docking. We took the collaborative helps, and that's why I talked to some of you, that those who have understanding in molecular docking can help us. And then we are also extended this TRPA1 channel, another channel, TRP channel, which is similar to previous one, V1. It regulates T cell responses. You can see CD69 and calciums are also down regulated if we inhibit TRPA1. And again, V1 in chikungunya. So you can understand that we are doing the basic immunology and then trying to extend it to immunosuppressive diseases like cancer or immunosuppressive drugs, as well as chikungunya infection as inflammatory model. And here we have shown that V1 is required for infection, and if we inhibit the TRP V1, which we have shown earlier with FJ in T cells, in macrophages, it is important for chikungunya virus infection. And this is the last one recently we have shown, because all the TRP channels are reported in different physiological purpose apart from their neurosensitive or thermosensitive drugs, but it is mostly shown for inflammation. This is the first time we have shown and found that in immune suppression, with cancerous microenvironment derived supernatant as well as immunosuppressive drugs, which is an autoimmune drug, FK506 tacrolimus, that V1 is important, and this is the cancer model, B16 melanoma, probably 40 and 50 years old model, still relevant, it highly metastatic and immunosuppressive. We found, and we also used the FK506, the tacrolimus, and there we found the same thing, that in activation it is going up, fine. But immune suppression also calcium is going up. So it is not the thing that only during activation calcium will go up. The details mechanism we do not know, by the way, at this moment. And this is the ongoing work. Currently we are looking for PLR4 in chikungunya infection, and nice interesting data are coming up. Our findings suggest that it is important for chikungunya infection, inflammation, and it also interact with some of the chikungunya viral protein. We are working with CMI as our lab works on that, immunosuppression, and then counter-regulatory. What happened to cancer like immune suppression and autoimmune or inflammatory diseases like chikungunya? Who is override others? So that we are looking for. And there is another interesting things with immune privilege. We know that there are different immune privilege organ in our body. Eye is an immune privilege organ. There also we get immune suppressive microenvironment. We are working on that. So I am at the end of my presentation. Still, I have 3 minutes time. So we are trying to unravel the mystery following the history of cell-mediated immunity, like infection immunity. And I'm ready for collaborations. Cancer immunity, because I was trained during my postdocs for cancer immunity. And also the basic T cell activations, immune activations, fundamental research without any disease in vitro. So thank you all the parties, the supports for our work, and they are our collaborator, the NICER, outside NICER. And this is our building. It was pretty old now, like now you can see a new shape. They are the small members. Don't get excited with so many members. There are only 4 PhD students right now, because in our lab we believe in excitement as well as peaceful environment. Those you can see the numbers is they are basically MSc students, and that's why I say that in NICER, where we have a double duty like teaching as well as research. So I feel excited and happy when we see the kids. So there are only 4 MSc PhD students, Tathagato, Khoma, Somlata, and Chandan here. They are the only 4 PhD students. Others are MSc students, and they are very creative. Thank you all for your nice timing, and I believe I have completed before time. Thank you.
Yeah. Thank you. Thank you very much. Yeah, now it's open for discussion. Is there any question from anyone? Okay. If there is no more question,
we can discuss later, because time is more important.
Yeah. If there is no more question, please thank the speaker again. Please join me to thank him.
So now I request the session chair to kindly come and present the memento to Professor. I request the session chair to kindly come and present the memento to Professor Subhashish.
Our next speaker is Professor O.E.C. Chakraborty from SINP. Our research interests include cell biology of diseases, organellar dynamics, cancer biology, and neurodegeneration. She has been awarded with Janaki Ammal National Women Bioscientist Award. She is a fellow of the National Academy of Sciences, India. Now I request Professor Oisi Chakraborty to give a talk. So it is for 25 minutes.
Yeah, I guess I'm audible. Good afternoon, everyone. It has been a wonderful stay so far, a very picturesque campus, a very well organized meeting. Thank you. Okay, so I have put this title very loosely. So organellar homeostasis. When we think about diseases, we think about the signaling cascades that are going wrong usually, and that's the first thing that comes to our mind, be it any of the signaling cascades, apoptosis, things like that. But we never think really closely about the gross architectural changes that happen. And when I say disease, it can be anything. It can be a neurodegenerative disease. It can be neuropathy. It can be cancer. It can be a disease due to viral infection, microbial infection, any of that. So during the course of the talk, I will tell you what I really mean by diseases. So when we look at a cell, it is basically doing it is tiptoeing on a tight rope. It is somehow balancing itself and maintaining the walk. But a perturbation actually forces it to fall. And that is what you see in the form of disease. And that can be any of what I have just talked about. In the interest of time and to ease our understanding, today I will deal with just two organelles. We do look at various other organelles also, but just two organelles is what I will talk about. This is the classical molecular biology of the cell image of the endoplasmic reticulum and the mitochondrion, where these organelles look actually stuck in space and time. But that is definitely not what is the situation inside a cell. When you look inside the cell, these organelles are continuously cross-talking with each other. And that crosstalk you can see as yellow patches, where the green is the endoplasmic reticulum, red is the mitochondrion. And this is a very normal cell. This is not an abnormal cell. Why do we choose these two organelles? These are the calcium stores of the cell. The endoplasmic reticulum and mitochondria buffer calcium inside the cell. In a normal cell, you have about 100 to 110 nanomolar of cytosolic calcium. Anything beyond that, even if you were to reach 120 nanomolar, that is detrimental for the cell. That denotes a diseased situation. And these two players are what are buffering the calcium inside the cell. So what happens if that calcium in the cytosol increases? So you think about the first thing is it's the second messenger, so a lot of signaling cascades will be going wrong. Yes, that does happen. But if you were to look at the structure of the endoplasmic reticulum, it expands. In a normal cell, the endoplasmic reticulum covers about 75% of the cell surface. But in a stressed cell, it can go till about 95% of the cellular surface. So it has really expanded. So endoplasmic reticulum, as the name suggests, is a network. Think about it. It is a network, or it's a fish net kind of a structure. You keep pulling it till the far reaches of the cell. Naturally, it breaks. You find these holes, these vacuoles, and this is what characterizes these. And if you were to look closely for okay, so how do we define these breakages? Here, if you see the red thread-like thing, it actually comes and joins to the adjacent structure at a within a finite period of time. And this breakage and making keeps happening. So the break and make keeps going on in a normal cell. But in a stressed situation, these breakages don't join up. So naturally, over time, what you have is that you end up with these big holes inside the cell. And these are characteristic of many diseases, neurodegenerative diseases, neuropathies, as well as in many cancers. So I am actually equating these two at all these at this level, because these structural changes do happen. Now let us look at the mitochondria. So the mitochondria, the classic textbook mitochondria, look very much kidney bean shaped, or rajma shaped, or sausage shaped, juicy, fat structures, oblong kind of structures. But that is not how they are in a normal cell. They are spread throughout the cell as threads. Those threads are really not very thin threads. They are very juicy, healthy looking threads spread throughout. But when there is stress, there is a perinuclear clustering. So if you were to look into the normal mitochondrial biogenesis, there is fusion of mitochondria and there is fission, which keeps occurring at all times, and that maintains the mitochondrial quality control, because that is the place where the membrane potential has to be well maintained, the ETC has to function properly, the calcium buffering has to take place, its own DNA has to be maintained, because this is an endosymbiotic situation, structure, where a bacteria historically got engulfed into a mammalian cell and lived for eternity, and that's how we have our mitochondria. So, and that's why it has its own DNA. So all that is well maintained because of this fission fusion cycle that keeps happening in the cell. But under stress, that is going wrong. So what happens when these mitochondria go wrong? They are actually autophagosed by a process of mitophagy, means mitochondrial autophagy. And here you can see that the autophagosomes are marked in green and the mitochondria is in red. In a normal cell, yes, autophagosed mitochondria are present, but their numbers, their proportion increases in a diseased, in a stressed situation. So here I'm loosely using the term stress, but we have seen this in neurodegeneration models, also in actually animal models as well as in cell culture models. So the endoplasmic reticulum goes wrong, the mitochondria go wrong. The junctions between these two organelles also go wrong. As you can see that in a normal situation, they are overlapping, cross-talking with each other. As you can see, multiple yellow patches being formed between the green endoplasmic reticulum and the red mitochondria. But the numbers increase, and this is a place where you see it almost all yellow. So that is really not a very healthy situation for the cell, and that is where you see that there is increased mitophagy. So how do we see what happens when there is increased mitophagy? So the mitochondria have two membranes, an outer membrane and an inner membrane. So here the outer membrane is marked in red and the inner material is in green. Here you can see that I'll just blow this up a little. you can very well see that the red is surrounding the green in a normal situation. But when there is high cytosolic calcium, the green and the red get decoupled. So the red is actually, the red outer membrane is lost from the inner, it stops surrounding the inner material, which is green over here, and the naked green comes out as mitoplasts. This is the first evidence in literature to show that we can generate mitoplasts in an ex vivo situation. Before that, people did not believe that mitoplasts did happen in under physiological stress or under pathological conditions. So we showed that yes, so you can see that the naked green and the numbers increase when the calcium stress goes even higher. So these are super resolution images, and here in the EM level, you can see that the inner membrane and the outer membrane are actually getting decoupled, and the endoplasmic reticulum is actually engulfing mitochondria. So here, the endoplasmic reticulum is, this is just a cartoon representation. If the endoplasmic reticulum was in green, the mitochondria here is present. And what you find is that these are actual images, 3D rendered. Okay, so these are actual confocal super resolution images which are 3D rendered. Here you can see the endoplasmic reticulum and the mitochondrial outer membrane in close proximity, but in this situation, the outer membrane is hardly present, the inner membrane and the endoplasmic reticulum are in close proximity. Not just that, you can see here that the endoplasmic reticulum forms these pockets inside which the mitochondria reside. So we have shown that we have actually identified a new process of autophagy where we call it reticulomitophagy. The reticulum comes from the endoplasmic reticulum, and the mitochondria are simultaneously turned over. And through lysosomal proteomics also, we have shown that yes, that is indeed the case. So now I move to the mitochondria. Okay, now I move to the mitochondria, where we know that with prolonged stress or excess of stress, we get these fissioned or fragmented mitochondria. But there is an intermediate situation where you get hyperfused or very long, extremely thin, well-connected mitochondria. So that is like a stopgap or a safe haven for overcoming stress. The organelle rather waits it out and thinks that, let me see if I can overcome the stress. Okay. Am I back? Yeah. So these are hyperfused mitochondria, which are long, thin structures. Here in the normal situation, you see that these are broken, but here they are not. So the hyperfusion can be because of two reasons. Either excess of fusion or hyperfusion can be because of excess of fusion or lack of fission. So what we find is that in this case, it is actually less of fission. As you can see, if you were to follow a normal mitochondrion, it breaks into two over a time period. But in the disease situation, this is actually from a condition of neuropathy, the Charcot-Marie-Tooth disease type 2, where you can see that in the same time frame, the mitochondrion is not breaking into two, and over time, this actually accumulates and gives the impression of hyperfused mitochondria. And why should hyperfusion be occurring? Mitochondrial breakage actually happens if this is a mitochondrion, the endoplasmic reticulum comes and wraps around it, and then many cytosolic adapters come and decorate on the endoplasmic reticulum, and then they break the two apart. Over here, what is going wrong is that the adapter molecule, which was to decorate the mitochondria, is actually getting degraded at a much faster rate in the diseased model than in the normal situation. And because of that, the less of the adapter protein is able to decorate the mitochondria. Through extensive MD simulations, in vitro, ex vivo studies, we have been able to show that the endoplasmic reticulum and mitochondrial junctions don't form as well, and those do not get decorated by these adapter proteins. Now I will switch gears and actually take you through what happened in the mitochondria in a cancer situation. So here, this is how a normal mitochondrion looks like. But if you were to look in GBM, glioblastoma multiforme, you find that the mitochondria don't look really normal. There is excess of crystallysis. There is a lot of fission also happening in these cancers. So are they abnormal mitochondria? No. So we go back to the players that we were looking at. And in an organoid culture model, we find that if this protein, the fission protein that I have talked about so far in a neuropathy situation, if that same fission protein is actually compromised, be it knockdown, knockout, what you find is that there is a gain of stemness. So these are the markers which go up when the fission marker is going down. Not just that, even the EMT is promoted, and as well as the wound healing capacity increases when this protein is compromised. So in want of time, I'm not showing you the ECAR OCR data, but what we have seen is that what happens when this protein gets compromised. We find that some of the players which help in tumorigenesis, like OCT4, ROC1, HIF1 alpha, all these are more nuclear localized. Why should that happen? So I will take you through our working hypothesis. The model that which we have proposed is that in a normal situation, this protein can interact with HIF1 alpha, and that interaction happens in the cytosol. And so a lot of HIF1 alpha is not actually translocating into the nucleus. But when you compromise this in a cancer situation, this can actually translocate into the nucleus. It can then sit on its binding site and cause promoter activation and absolute downstream signaling by the OCT4 and ROC1, as we see in our situation. So with the same players, with the same set of proteins which were actually going wrong, which were regulating situations in neurodegeneration, neuropathy. Practically the same players participate in cancer. It's just that we do not really appreciate, do not have an understanding of these so far. So the take-home message is the endoplasmic reticulum or the mitochondria or the endoplasmic reticular mitochondrial dynamics, they can regulate the ER turnover, mitochondrial turnover, ER mitochondria turnover simultaneously, the mitochondrial dynamics. Now, I will take the last 1 minute to talk to you about another important thing which is present in the mitochondrion, that is the mitochondrial DNA. The mitochondrial DNA actually encodes for just 13 polypeptides. There are about 37 genes, but only 13 polypeptides are encoded in the mitochondria. So, when in a normal cell, the mitochondrial DNA is well preserved inside. When there is destabilization, be it any situation, even in cancers, we find that this DNA can escape from inside the mitochondria. Why should that be of any significance? In this COVID-19 time, it becomes very important because the escaped DNA, the extra mitochondrial DNA can actually activate the C-gasting pathway and activate innate immunity. We see a ton of immune response happening in many patients with increasing mortality. Why should mitochondrial DNA be again important? Because when it is almost used in case, like a diagnostic tool in many cases where the circulating DNA is actually looked for in many cancers. The serum DNA, that is not always of nuclear origin. It has a mitochondrial origin too. So how does this DNA come out from the mitochondria is again one question that we are trying to address. Now, I just told you that the mitochondria encodes for only 13 polypeptides. So, and there is naturally, so if 13 polypeptides are encoded, there has to be some problem going on in encoding, in translation, transcription, things like that. And an unfolded response at the mitochondrion is also possible, and it is absolutely evident. So that unfolded protein response actually crosstalks with the nuclear unfolded protein response. And there is a continuous interaction between these two responses to actually elicit the final outcome, what the cell sees, whether it is apoptosis, whether the cell proliferates, or whether the cell tries to re-establish its homeostasis. So these are the primary players behind the scene. I have talk to you about the work of this gentleman, this individual here, 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 and here. The rest, I did not get time. These are my collaborators. This work has been primarily funded by intramural funding from DAE with partial funds from the other agencies. Now, I would like to end my talk with actually one thing, that being in a physics institute, we have been doing biophysical sciences and biological sciences since time immemorial, actually. And naturally, this was very well recognized by Professor Meghnad Saha, and our biophysical sciences actually date back till the time of Azadi. Thank you very much for your time and attention.
The talk is now open for discussions. Yeah.
Hi, Oisi.
Yes.
Wonderful talk. Actually, I was wondering, in cancer cells, when you see the mitochondria, EM, it shows that inner mitochondrial membrane Christie is disrupted, right?
Yes. But now the question would be, how does ETC happen? How does the generation of ATP, etc., or energy production, all that happens? So just a while ago, I was talking to Prasanna, and I was actually, it's just a hypothesis. I'm speaking my mind aloud, that if you have about 100 mitochondria in a cell, in a normal cell, and they work at 99% efficiency, they cannot be working at 100% efficiency, 98 to 99% efficiency, whatever you get, versus a cancer cell where there is fission, you have about 150 mitochondria now, but they are working at about 80 to 85% efficiency, but still
So
the output
it's a
would be better.
number is more.
Yes.
So no, my question is, yeah, apart from that, it's so looks like it's how DRP1 is actually rescuing some of it.
Well, we just wait for our paper now. Okay, we are really trying to deal with that. It is something quite confounding, and especially with the lot of controversy now happening with the HIF1 alpha. In the recent past, there is a huge amount of problems which have come up. I would not do, I mean, really be vociferous about it in an open podium, but we have to really see what is going on. One of the hypotheses that we are proposing is that the interactions are what is going wrong. And here also, in the cancer also, there are actually multiple populations of cell. One would be the brain tumor initiating cells, the BTICs. Then there would be the other, which are more differentiated like fate. So where does DRP actually work, in the BTICs or in the more differentiated like tumor cells? We are still trying to understand that, or they help in the conversion. We are trying to address that. Yes, we have checked for that. OPA1, we have checked for the entire battery of proteins, all the fission fusion factors, the various adapters. We do not see too much change in those levels. So that also becomes quite confounding for us. Why should those factors not be changing as much? Yes, it is.
You can go to the next person. Yeah.
Wonderful. Can you little bit elaborate on the UPR between the mitochondrial UPR response and the
Okay, the UPR at the mitochondria is actually at various levels. Mitochondrial, so now all mitochondria, most of the mitochondrial proteins have to be imported into the mitochondria. There are about 5 or 6 import pathways. It's very complicated. So anything can go wrong in these import pathways. Okay, and that actually causes UPR at the mitochondria, and that's the understanding so far. Now, if that one of those don't get imported, naturally that gets accumulated in the cytosol. Now that protein which is getting accumulated in the cytosol was actually translated from the nuclear pathway. So that is causing a feedback into the nuclear pathway. When that feeds into the nuclear transcription translation pathway, then the cell realizes that something is wrong, not just at the mitochondrion, I should also become cautious, and it generates a nuclear UPR. So that's how the crosstalk happens at a very basic, like a kindergarten level is.
So for the mitochondrial UPR, does one see aggregation of proteins?
You do see aggregation of proteins. You see aggregation of proteins in the cytosol. We have a study going on. I have not gone into the details of it. We see proteins accumulating in the cytosol. We see proteins accumulating at the outer membrane. We see proteins accumulating in the intermembrane space in the IMS. So these are the various places where proteins can accumulate. Yeah.
Now you can.
Yeah, Pankaj.
So I was wondering what is actually happening when this endoplasmic reticulum is invading the mitochondria. So what is happening with this calcium? So you said something about the calcium.
The calcium buffering occurs when the mitochondria, there are these junctions. There are various types of junctions, MAMS, RMS, various types of junctions having very different protein, protein composition as well as structural differences. So these are the junctions where calcium buffering happens. We have data where we see that if you were to just give a little bit of stress, you can see a lot of calcium coming out from the endoplasmic reticulum going into the mitochondria and vice versa. So they try to buffer with each other. If that does not happen, then the ryanodine receptors, IP3Rs, all these come into picture and throw the calcium out into the cytosol. There is one question from a student or a, oh yeah, please. Yeah.
Oishi, thank you. It's a wonderful talk. I mean, maybe my question is a little bit off the what you have said. It's more. So there is in the cancer area there is also a concept erupting now called migrazomes. So which actually and these bodies are transfer mitochondria from one cancer cell to another. So do you have such experiences, or can
We
you share
don't
anything
have,
on
but
this?
that is one thing we have been thinking to see if you can transfer these, have an because that does happen. Yes, we have knowledge that these junctions are formed and they are like thin, really thin tubes and
Cell-to-cell
things.
communication.
Yes, cell horizontal transfer is really what happens, but we don't have any expertise on it, but that's
Those are incidences to catch, means
yes,
the resolving those kind of situations in a dynamic
absolutely.
scenario.
It would be wonderful to really have that see that in living cell.
It's a wonderful talk. See, I have just a general question. Might be wrong. See, inside the cell, you have sodium, potassium, calcium. So how actually this, and it balances the ionic potential inside the cell. So how it is very specific to from that kind of situation, how it is selectively identifying that there is a excess calcium or deficiency in calcium.
How the cell understands that?
Yes, because
Okay.
it actually there is a ionic potential due to several ions.
Yes, yes. So there are various calcium. So I did not show our data. We do have a work on that also. How does the cell actually sense that I have more calcium? There are various calcium binding proteins in the cell. These calcium binding proteins can be in the apo form where it is unbound, and they are in the holo form where they are bound to calcium. Usually about 2 to 4 calcium bound states are there. Okay, so that structural change happens in these proteins when they bind to calcium. When these proteins undergo structural changes, then their protein-protein interaction partners change. If one protein is calcium bound, if it were interacting with, if in the unbound state it was interacting with ABC, in the bound state it probably interacts with PQR. So that's where the sensing actually comes into picture.
I was actually wondering about the structure of the endoplasmic reticulum. So you said that over time there is a destruction of edges and then there's a remaking of them. So if one looks at the entire net in one go at some instant in time, how does the structure change? Is it like synchronized over different regions or?
It is, I do not have a direct answer to it. I don't know if it is synchronized or not, but what I can say is that the entire endoplasmic, if you were to look at these hexagonal kind of, I mean, holes that make the entire network, they are actually formed by Y junctions. These Y junctions are made and formed by multiple proteins. At least about 10 to 15 proteins are responsible for making and holding this Y junction. So when the endoplasmic reticulum starts breaking, these Y junctions start breaking. The proteins do not come and hold these together, or they don't form properly. So that's what happens, and that's when the structure falls apart.
I am audible?
Yes.
Yeah. So this is from a structural point of view. I was just wondering, and the talk was beautiful. So regarding the structure of the ER that you are talking about, so is it intercalated with the cytoskeleton?
Absolutely, yes. When there is a calcium surge, the actin network also changes, and that can be measured in live cells, and that can be actually coupled with changes which are happening at the endoplasmic reticulum.
Okay. So this, okay, that's fine. And then about this mitochondrial destruction that you're talking about, that destruction is happening because of imbalances, ion imbalances between the mitochondrial membrane potential within the mitochondria and outside that of the mitochondria. Is it happening due to that?
It is happening, yes, absolutely. Membrane potential imbalance will happen, but before that, the outer membrane sloughs off, it separates out.
Is it because due to imbalances in the outer membrane?
The proteins in the outer membrane get plucked out from the outer membrane and get degraded. It is not, the initial trigger was not imbalance at the, it is mostly to do with degradation of selective proteins. So if there is a membrane and you keep plucking things out from there, it will get destabilized.
Of the membrane.
Yes, so that is what happens. Yeah.
Sure.
Thanks, ma'am, for a very nice talk.
Okay.
Just I want to ask that whether only cytosolic calcium ions are responsible for causing all this degradation of mitochondria, or there are any other stress factors?
Lot of stress factors. To just make the talk very simple and to elicit lots of questions from you, I talked only about the calcium surge.
Can you just list some few?
If you have protein aggregates present, you will have a similar response. Like unfolded proteins, if or like misfolded proteins which did not get degraded by the proteasomal machinery, you will have a similar response. Yeah. Thank you very much.
Maybe during lunch you can discuss. So please join me to thank Professor C.
Now I request the session chair to kindly present a memento to Professor Oishima.
To the third lecture of the session, Professor Tirumala Kumar Chaudhary from NISHAR, Bhubaneswar. His research interest, structural biology. his research interest, structural biology of viral molecular machines. I invite Professor Tirumala Chaudhary. Yeah.
Yeah. While the slides are coming up, thank you for having us here, RRCat, for being such a wonderful host, and the place itself is such an exciting one. So, my lab interest is, as the title says, we are keen on understanding protein-protein interactions and conformational dynamics of viral protein complexes. I will elaborate a little bit on what do I mean by that. So, what we are looking at is protein complexes involved in different aspects of viral biology. The current focus being one that envelope viruses entry, how do they enter, and what proteins are present on the viral surface that would facilitate this entry. And another aspect that we are interested in the viral biology is how these viruses, once they are inside and replicating, what protein complexes take part in such kind of mechanisms. And of course, the genomic packaging is another. I'll be briefly talking about these two aspects. I'll keep this for the next talk. Probably this slide is working. Oh, okay. Okay. So to briefly talk about what interests us in terms of viral entry and what are we trying to understand is, as these viruses now doesn't need any interaction, if at all. SARS and influenza, which causes flu, it's familiar to all of us now. But chikungunya, thanks to the earlier speaker, Dr. Subhashish has already spoken about, but this is a mosquito transmitted virus. But the reason why we are interested in these three viral groups at the current point is all of them have in their structure a lipid bilayer envelope that covers both the nucleic acid genome and the other proteins involved. And what is the most fascinating thing about that is this lipid envelope has proteins and protein complexes involved, embedded into that membrane, which irrespective of whether you're looking at SARS-CoV-2 or influenza, or chikungunya, or for that matter, a bunch of different other envelope viruses, they all have these protein complexes embedded in this, which look and function more or less the same way. So it's a universal mechanism that operates in these proteins to facilitate the viral entry. And if you look at it, obviously for the envelope virus, unless the first step in the viral biology is for this virus to enter inside the cell, and only then it can be biologically active, or you can actually consider it as a biologically active molecule only once inside. So there are two steps, critical steps that are involved in envelope virus entry. One, of course, it has to know which cell to enter. So the cell surface receptor is what would determine, and this protein's obvious choice for virus would be the proteins present on the surface, which, since if you look at it, all of them are looking like spike-like projections from the viral surface. So that's why this word S protein or spike-like proteins. Though you may be familiar with SARS-CoV-2 S protein, even other ones, we call them as spike proteins or envelope proteins for various reasons. But the spike proteins do two things. These are the obvious choices for the viruses to start their biology. One is to bind specifically to a virus, the cell surface receptor, so that the virus knows that this is the cell that I can infect and enter inside. So that's the first gateway. Once the receptor binding is done, so that leads to a signaling cascade which would allow this virus, in most of the cases, including SARS-CoV-2, for the sake of time, I'm not going into the different pathways these viruses can take, but most of them do take this pathway of getting endocytosed, and then once the endosome gets enough acidified, I'll talk about what this enough acidification means, but once the endosome gets acidified through proton pumps, pumping protons into it, then there is a second critical step that ensues. That is, the viral membrane, whatever that is there, which is protective in role outside the cell, now has to fuse with this endosome vesicle membrane so that the viral contents can come out, and only then the virus can start its biology. It's a simple theme that almost all viruses, especially the enveloped viruses, many of the human pathogens or animal pathogens fall under this category of enveloped viruses. So obvious choices being, you can see why we are working on that. But our interest is primarily on these steps, receptor binding and this membrane fusion. And the protein complexes, how do they perform these jobs? Why am I emphasizing on this is, if you think of it, this is more of a mechanical job, like pulling this membrane and this membrane together and fusing them, or this triggering a signaling cascade and doing this. All this is happening when the virus is not metabolically active yet. What does it mean is, there is no ATP source for it. There is no signaling cascade interactions that are possible for these proteins, but they act like machines. That's why our title, the title says that understanding the conformational dynamics of these machines. Okay, so that is what interests us. So let me give you an example of what this means. What is this conformational dynamics? Why it is important for us to understand on how these machines are working. And this applies for both replication complexes, as well as packaging complexes, as well as the entry complexes. So the classical example of such kind of proteins is influenza hemagglutinin, because that's the first structure of any viral envelope protein that we know of. If you look at it, and if I zoom in on one of those small projections from an EM image, if you look at it and fit the crystal structure that is available with us, and you would see that it is like a small projection up, or vertically going up from the viral membrane, and you see like a mushroom-like structure where there is a stalk of the mushroom and there is a bulbous head of the mushroom. And for influenza, this bulbous head, which I'm showing in this gray color one here, that is where the receptor would bind. And influenza, like SARS, is an upper respiratory tract infection, which means it would bind to some of these sugar moieties present in your mucus layer that would allow them to enter into the respiratory tract cells. Now, the second half, which is looking like more stalk-like, is the HA2. That would be doing the second step, the membrane fusion part. Now, what is interesting is, in the context of these proteins have to work in the absence of any of those metabolic signals or ATP energy source, none of them being there, this protein spontaneously has to undergo a conformational switch from this to this. What is missing in this? Obviously, this gray colored one. So obviously, the receptor binding should allow this molecule to shed this molecule away and then undergo a dramatic conformational change where this only short stalk should extend like a long extending one, so that this part can go, say if this is the viral one, so it can go all the way up to the endosomal membrane and then do a mechanical pulling action to do this job. So we are interested in this pre-fusion to post-fusion, what I would be referring to them, is what our interest is. Because for the obvious reason that this is happening spontaneously, and anyone who understands protein folding here would know that you would expect these proteins to be in a energetically favorable state. That means they are already at a free energy minima state. That's how you would try to keep entire system of whatever proteins or different molecules, whatever that you're talking about. That's what you would see as a stable conformation. But what is the peculiarity of this kind of proteins is they do keep up in a metastable state and then switch to a more stable state once a specific trigger comes. The specific trigger in this case is receptor binding and the acidic pH of the endosome. So just by sensing these two, these proteins have to switch from one conformation to the other conformation. And that's not I mean, if it is happening in one protein of one particular virus, probably would not have got excited about it that much. But what is interesting about it, if you look at any of these proteins, may it be SARS-CoV-2S, influenza HA, or chikungunya E1, E2, all of them show more or less the same kind of conformational transitions during entry. So that is what is exciting to us. So that is what we try to look at. So ironically, ever since like last 30 years, we know these structures, but how do these molecules switch from to here, and what is the trigger is still remained an elusive topic for many of us to explore. So that is something that we are excited about. So this schematic we can skip because this is basically reiterating the same thing, but with an exception for chikungunya and chikungunya-like proteins, where especially chikungunya, dengue, and hepatitis C, the mechanism is the same, but the structural features what you would see in influenza, SARS-CoV-2, and this chikungunya and dengue are slightly different. So at least some amount of understanding is there for SARS-CoV-2S and influenza and similar proteins, but for with respect to chikungunya and dengue, this class of proteins, we don't have absolutely no information, especially how the receptor binding would shed away this, and so that the second aspect of acidic pH triggered membrane fusion can happen, that is completely lacking. So that's something that we are excited about. That's the theme that we are looking at. And before I go into the specific case of chikungunya, you may wonder why, how do I get help by studying these conformational changes? Yes, the protein is there and it's switching from one to the other as a fundamental concept of how protein folding happens and structure function relationships. It's wonderful. But what does it mean in terms of significance, in terms of application to viral therapeutics? So what is interesting is, since this mechanism is conserved across all, if you understand the conformations that has to on the viral surface and the intermediate conformation and how that information, the conformation is changing, you can block the viral entry just by stopping this conformational change itself. So that is the theme that many who are working on viral entry are excited about. So the classical example of this being a successful story comes from HIV. HIV treatment, probably many of you know that you do a combinatorial therapy for HIV because HIV being a virus can mutate very rapidly, and whatever drug that you develop, may it be for your polymerase, may it be for other proteins, it would quickly come up with a variant which would inhibit that resistance or overcome or come up with a resistance that would make the drug non-useful, right? So unless you stop the virus from entering, there is no possibility of a mutation happening because the virus did not get a chance to start its biology. So that has been the successful story for HIV. HIV, one small peptide, which is complementary to the structure of the HIV spike protein, is what is the only successful drug where the number of resistance mutations are less. It's not completely off, but if you compare all the combinatorial drugs, the T20 peptide, what we call it as, that peptide is the most successful strategy, and that never changed in the combinatorial therapy. Rest of the drugs keep changing. So goal is, for those of us who are interested in understanding this conformational studies, to come up with one molecule where you would be able to prevent a multitude of viruses, may it be for SARS, may it be for influenza, may it be for any other viruses. One drug, at least for a bunch of different viruses, you take one tablet, you are cured. Not only symptomatic relief, but cured of these viruses. So that's the kind of philosophy that drives us to understand this. So I'll come back to chikungunya, where if you look at it, so this is once again, that's one cryo reconstruction of the whole molecule, the particle. And on the surface, you would see these small spike-like projections. If I show it in the vertical axis, this is how it would look like. And from the top view, that's how it is. What is interesting in this for us is this what I'm representing here in this golden color or golden brown color is the receptor binding protein. Let's not worry about the names, but I'll be referring to them with the colors. So that golden color and this gray color are the two steps. The first step, receptor binding. The second step for the low pH triggered membrane to membrane fusion. And for this also, if you see, receptor binding domain is off. After the post fusion, membrane fusion, only 3 E1s or the 3 membrane fusion parts come together and make a homotrimer. Okay. Now, interesting thing is, I told you that chikungunya and dengue, these proteins are slightly different from the structure because there in HA or HIV, GP120 or other Ebola and all those bunch of different viruses, what makes this conformational change, at least we can explain from the structures. Because we have long coiled coils, one helix coiling onto the other helix as a structural feature, we can understand it. But for these proteins, we absolutely don't know what exactly is happening. So what we started with is we wanted to look at what is the conformational path that this protein would take from this state to this state to perform this membrane fusion and entry process. So to start with, briefly this I have put up for, this is not from my students' DC committee presentation, but I intentionally put it so that to give you a sense of the expertise that we have built up over the period of time, so that if anyone would be interested, especially this aspect, we have expertise in making these protein complexes in insect cells. And you would have obviously noticed by this time why it is important for membrane complexes or protein-protein complexes. You need this kind of post-translational modifications to go through the secretory pathway. Insect cells is one such one which is, of course, for viral proteins, there is no other option for us. That complex, what I showed you in the last slide, comes only if I express them in a suitable system. Insect cells is one such system. But now we are capable of making such complexes and purifying them, and we can help you if you would need such kind of an expertise, and we'll be more than happy to help you with that. And then we also have expertise in single photon electron microscopy, and nicer we have a new GeoLF 200S cryo-EM facility that is being installed, and it's almost up and running now. So that is another area that we would be more than happy to help you if anyone would be interested in. Okay. So other tools, a bunch of other tools are there, but I will not go into those. We also are excited about this. We have expertise in developing, for those of you who are into transfections for primary cell lines, you know how much it is important for lentiviral pseudovariant systems or VSVG kind of pseudovariant systems for transfections. We have in our lab built up a much easier lentiviral, pseudovariant system, which is a baculovirus one, which would involve only transfecting one BACMID, unlike your lentiviral systems or VSVG systems, where you have to have multitude of plasmids. So we will be happy to share if anyone would be interested to look at it. So I'll quickly go through the data. I will not bore you with too much of details, but the first thing that we didn't know about chikungunya is what is the receptor and where does it bind when we started it. Of course, later on, several other groups have showed it, but we found out that chikungunya can bind to this heparan sulfate, a small moiety, sugar moiety present on several proteins on the mucous layer of the epithelial cells. So those would be the primary targets for binding, and that can be used. And we found out that within the trimeric spike, between one receptor binding domain and other receptor binding domain, that's where a charged pocket is where this would bind. I'll skip some of these details, but we found out that the heparan sulfate binding moiety, what we found in these proteins is a different one, which has not the motif. What we found is completely different than what is reported in the literature so far. And then we later on actually biochemically established that E2, the protein that is purified protein as well, can bind to and heparan sulfate with a good affinity and through charge interactions. So, and when we remove some of these residues, charged residues, this arginine, lysine, when we remove it because of the heparan sulfate moieties and arginine moieties. So that we can abrogate the binding, and that gave us the confidence that this is a right receptor to look at. So, but what we are interested in is in this conformational dynamics. So what we did is we started with taking an in silico approach, because there is no easier way for us to really take this whole complex and look at it, the resolution what we could, or at least what we wish for in any other method. We started with molecular dynamic simulations on it, and our simulations, when the heparan sulfate is binding, what it showed up is that the heparan sulfate binds here, but the domain motions, large scale domain motions, is observed in this particular area, the third domain here. So that we corroborated with the Fred studies, showing that this domains actually, the interdomain distance is what is changing when we add heparan sulfate with it. And what it means is, to make the story short, so binding of heparan sulfate in this would, if I overlap it with, I'm not sure if you can clearly see the gray color here, but if you look at the E2 before heparan sulfate and after heparan sulfate binding and overlap with the heterodimer structure, see this domain is kind of pushing this other protein which would be responsible for the fusion. So this, based on that, this is what we proposed, and even now, even for influenza, we don't know how the receptor binding, that mushroom head, the gray color one that I showed in the earlier slides, that how does it dissociate, whether it completely falls apart, what triggers this dissociation is not known. So we kind of gave a first indication of how this receptor binding domain motions can be triggered after receptor binding is what we showed. Am I already done with? Fine. So I'm done now. So then we also looked at acidic pH sensors. So histidine residues and charge residues in there is what we looked at, because those are the likely targets. So, and we by various methods, both in silico and in vitro methods and electron microscopy methods, we could show that a short, I will skip to this, a short hinge region which is kind of responding to the acidic pH change by changing the ionization of those charged residues, that would break up a salt bridge, and that would lead to this extended conformation. We still don't know the intermediates, like how this would be extending into it, but we are currently working on it. So as a continuation of that story, another PhD student who will be presenting his poster, Naresh, would be presenting it in the poster 38. I'll briefly explain in one sentence. So we are also looking at how this, eventually this protein has to insert into the host membrane and then pull it. So what are the kind of conformational changes that would happen in this? What sequence and structural determinants are there that would allow this to insert only into a particular depth and then pull it? So this is something that we are excited about. I'll slightly switch gears and talk about the other aspect, that replication complexes that I mentioned about. So flaviviruses, especially the dengue doesn't need an introduction here, but so that RNA is replicates inside a cell, inside if you look at it. So we are talking about immune escape. So viruses have mastered these mechanisms of how to not being sensed. Any double-strand RNA, you would obviously know that there are a lot of innate immune responses that would stop it and destroy those double-strand RNAs. But for viruses which have to replicate in the cytoplasm, especially RNA viruses, dengue being one of those, they have developed these mechanisms where they make a small vesicle as an invagination into the ER membrane, and that is where all the RNA and viral RNA and viral proteins come together and make a large complex, and these through these protein complexes is what the replication happens. We are specifically interested, I'll skip some of these details, but we are specifically interested in a couple of these proteins interactions, NS3 and NS5, because these are the two proteins that are absolutely important, and their interaction is absolutely important for virus replication. So once again, there are so many drugs that would block this interaction NS3 and NS5, and that would stop the viral replication and viral infection. So along with these two proteins, there is another protein called as NS4B. This is the focus that we are currently having. So the reason why we are interested in NS3 is internal part of it is a protease, and the remaining part acts as a helicase. So it's a dual function protein. the canonical serine protease motif here, domain here, and the helicase canonical SF2 class of RNA helicase motif once. So that we could show that that this interaction between NS3 and NS4 is critical, and through various methods, what would be the allosteric mechanism, how this interaction would be triggering this in terms of the increasing the helicase activity and optimizing the helicase activity is something that we have looked at. So I will not go into all these details, but some of this protease part is presented as a poster by Simanchal in the poster session in the afternoon. So protease interactions I will not be touching upon. So with that I will come to the last slide and the most important slide. So the work that is presented here is done by one of my earlier PhD students, Vivek, and Satyamurthy, who is writing up his thesis right away and right now, and some of the extension of the work will be presented in the posters. So other members are both MSc and PhD students in the lab, and funding from DAE and DST and DBT. I would be happy to take any questions.
Yeah, the talk is now open for discussion.
Yeah, it's an excellent talk. I was wondering about the VLPs. We know that they can be used for delivery agents of these genes and all.
Right.
Are you looking at possibilities of using VLPs for delivering something other than genes, like drugs and something?
So the reason why we started with this, the pseudovariant system that I told you about that we developed in-house, we took a baculovirus, which is an insect virus, and it's been used as a pesticide in agriculture for quite some time. So it's totally safe on this. So then we modified some of the genome of the DACMate to incorporate a GFP into it, and then also be able to take out whatever that we need and incorporate it into it through a transposition system. So that gave us this advantage of, like, initially when we started it, we wanted to use it for viral entry, but then once SARS-CoV-2, we could pseudotype it. So we wanted to propose it as a vaccine candidate, because baculoviruses, S protein is an ideal candidate. The cost of production, the safety, and all of that. Unfortunately, it didn't take off the way we expected it, but we have a reporter system in there, and we have the amenability to modify the genome in a very simple way, in whatever way we want. So both reporting as well as for delivering, we can use it.
Okay, fine. Thank you.
So I have a question regarding the virus entry to the host cell. So you said the spike protein actually undergoes the fusion conformational change. So what about DNA viruses? Is this kind of mechanism conserved?
You would see that if you are talking about HPV or herpes simplex viruses, all of those. So those, if the mechanism wise, it is pretty much the same. Okay, there is no change. But the protein complexes there are much more complex. But if you look at this conformational switch, what we are talking about, that is conserved. That would be following the same thing. In fact, there are studies earlier, not from because there is some amount of membrane requirement also, membrane structure, how it should be for this fusion protein to insert it also. So from that angle, people have tried small molecules, like simple rhodamine, which would partition into the membrane and changes its fluidity, has been shown to stop both enveloped and non-enveloped rotavirus, herpes, some of the DNA viruses, some of the RNA viruses. One rhodamine derived molecule from the rhodamine could stop 18 different groups of viruses. Of course, that is more in the test tube level, but for various reasons, it could not really take off. But that kind of concept is what we are looking at. So it can apply for both DNA viruses as well as RNA.
So nice talk. So I have a question. So you mentioned that the pre-fusion and the post-fusion form, it is roughly a metastable kind of a conformation, right? So
because that's the best explanation that we can give.
So if the pre-fusion form is so unstable, do you think it is stable enough to for the experiments to I mean, for that long, is it experiment like can it last, or you are putting some stabilizing mutations to stabilize the prefusion form as shown in RSV virus?
So your question summed up all the difficulties what we have in studying them. So yeah, these proteins, one, because they have this transmembrane, small parts of transmembrane region, so we cannot work with those. We have to work with only the outer sides, and the stability, and the reason why we cannot do a simple bacterial expression or many other methods of this is the metastable state is kept only when this protein-protein complex is there. The moment I take that receptor binding protein, the gray color part which I was showing, or the orange colored one, then this protein adopts a conformation which I don't have absolutely no control. Whatever studies that I do on it, I'm not sure whether I'm doing it in the right conformation, what I would expect it on the viral surface. But that's the difficulty that we have with it. So we have to work with those protein complexes. And of course, when you work with the complex, the flexibility that you have to understand these domain motions, what probes you can probe it with, that reduces quite a bit. So cryo-EM is something that we are excited about, and our field is going in that direction. So we are hoping that cryo-EM imaging on these molecules and then single molecule reconstruction on those would help us to understand it a little bit more.
Okay, and last one is like you mentioned that these are glycoproteins, right? They have
Yeah,
glycans.
yeah.
So SF9, is it a good enough host to preserve the integrity of the glycan chains?
Once again, that's the best choice and easier choice that we have. But ideally, you should say some of those proteins, you cannot express them in insect cells. So for example, HIV. HIV GP120, if I do it in insect cells, there is no guarantee for me that it would be the same as this. So there I have to depend on either CHO or Viro, some of the mammalian cells where the glycosylation pattern would be more or less matching to what would be there on the virion cell. And the glycosylation, say from the same cell, if 10 particles are out, glycosylation pattern on one viral particle to the other viral particle can be different. There is some amount of heterogeneity there. But that's the experimental difficulty. But these are the easier systems what we can work with. But you are right that you have to be cautious about and learn about which one system would be a better option.
Yeah, I have a naive question. I don't understand much of it, but still. So you mentioned that the virus is not metabolically active when it is internalizing, right?
Yeah, yeah, until it escapes the endosome. Yeah.
Correct. So there has to be a humongous amount of force to deform the membrane per se. What provides that force?
So yeah, so in the schematics we show them like nice spheres.
No, but that
That
cannot
is not the case. Say influenza is pleomorphic, SARS is pleomorphic. You see these images in a control system. Because most of the EM images what you see are isolated viral particles of a particular density and a particular overall shape. In that context, those are spherical. But if you look at in a tissue system, in an infected cell, if you take an image, you would see this membrane to be like expanding and very pleomorphic structures. Now coming to the second part of your question, how does that membrane getting? So each virus has different mechanisms. One, the chikungunya that I showed you, which look more or less spherical, and the dengue also, because the proteins that are embedded in this protein also follow kind of a lattice structure. So they're kind of compressing it back, yeah, collapsing the membrane
So
back onto
the bar body proteins are the bar kind of proteins. They are
some of these proteins can sense the curvature,
Correct.
so they would work more or less the same way as the bar domain proteins. Yeah.
I'm sure this must have been thought about by others, but if it is a pH sensing, the protonation state of any amino acid that changes in the environment should trigger this.
So yeah, that's actually part of this work. For the lack of time, I couldn't really talk about it much, but you're right. So the obvious choice in that scenario would be the histidines, because neutral to low pH, that histidine protonation changes is what you would expect. So that's what is. So what was thought to be, I mean, this is once again from an extension from the influenza HA protein. So the histidines were the primary target that people thought, okay, this, and it is proved to be true for influenza because some critical residues where you could sense how the protonation changes would happen on the histidine by protonation to the singly protonated state. And then the what interactions would change based on the protonation state. So influenza, that guess turned out to be wonderful. And if you change that histidine to something, the molecule becomes inactive. We started with the same guess. We looked at all the histidines and from the biochemistry, whatever is shown that, okay, these histidines are probably important or not. It turns out that in our case, this particular protein case, none of those histidines turned out to be, and we started with the that I think I did not have that PK states, but we titrated this protein to pH 5.5 in silico and showed, I mean, looked at what are the protonation changes change. What turned out to be is like there's a salt bridge here, which is unusual candidate for this, but that is showing rapid ionization changes. We are trying to look at how the environment of this is changing that could trigger. But for in silico, it is too big of a molecule for with the computational power what we have. It is not really helping us to have the whole complex and study it. But yeah, you are right that that's the kind of mechanism that operates.
Okay, one last question. Anybody? You was asking.
No, sir. It's a very naive question again.
Yeah, please.
So the thing is there any kind of functional lipids which help or accelerates the membrane fusion?
From I wouldn't call a single. I would not be able to put a one indicator on any particular lipid, but raft-like rigid structures, especially cholesterol-rich domains, were shown to be important for when the rafts field was like booming. Everything is connected to rafts, right? So, and it is true that HIV and some of those prefer those domains, cholesterol-rich domains in the membrane where the insertion of the fusion loops can happen more easily. And if you take out some of the cholesterol, deplete the cholesterol, the fusion efficiency, at least in vitro, is significantly reduced. But for would it operate for all viruses? And there is a lot of dilemma within the field.
But does that change when you try to purify it in an insect cell or something like?
For purification aspect, no. But this, if you have a system, organ system or a organelle system, membrane system where a lot of curvatures are there and lysolipids are there, there you would expect this protein to be having a different conformation. We haven't checked it, but others have shown that it would preferably, if you have a membrane where the composition is more of lysozyme so that the inverted pyramid kind of structures can happen and the sharp curvatures can come, the protein would go and sit there and won't move from there.
Thank you.
Thank you.
Now I request the session chair to kindly present a memento to Professor Tirumala.
Now we move to the last talk of this session. Professor Rohan Khadilkar from ACTRAC. His research interests include Drosophila model of cancer, stem cell, niche interactions, tissue homeostasis during development and disease conditions. He is awarded with INSA Young Scientist Award, Hargobind Khurana Innovative Young Biotechnology Award, Ramalingo Swami Re-Entry Fellowship, and EMBO Research Fellowship. So with this I invite the speaker. You have 25 minutes.
Good afternoon, everyone. At the outset, I would like to thank HBNI for conceptualizing this first of its kind theme meeting that focuses on life sciences, and RRCAT for executing it wonderfully. We're very happy to be on campus and be at this meeting, and we look forward to interactions, future interactions with other HBNI institutes. So, ours is a young lab. We started 2 years ago, just before the pandemic, and the work that I'm going to present today has been done for the past 1.5 years. And our lab works on the Drosophila system. We work on Drosophila as a model organism, and we also plan to translate it into the mammalian system by looking at not only cell culture-based models, but also patient samples, especially some of the cancer models that we are modeling in flies. We plan to translate those into the mammalian system. So Drosophila is a powerful model organism, firstly because of the power of genetics that it offers. You can answer a number of biological questions using the system. It has a short life cycle. It's easy to culture and grow. It grows in around 2 weeks. You can propagate it fairly simply. The genome is sequenced and well annotated. And there are a number of gene manipulation technologies that are sophisticated enough that allow you to manipulate the genes in a spatial and a temporal manner. The genes are conserved. Name it, most of the pathways that people look at in the mammalian system have actually originated from the flies. The studies, the genetic screens have been done in flies. That's why you would find the funny names that lead to the mutant phenotypes and the names of the pathways and the molecules. In vivo analysis is easily tractable. You can look at different organ systems. You can look at inter-organ communication and crosstalk. That helps in understanding and in a coherent in vivo scenario in the organism. So the lab is currently working on 3 different aspects currently. We are working on understanding how stem cell and tissue homeostasis is affected in a disease scenario versus a physiological scenario, wherein we are focusing mostly on the hematopoietic system. That is where we are trying to build the Drosophila model of leukemia, wherein we use the human mutant transgenes, and we are trying to propagate that and induce those in the flies to see whether the leukemia phenotype is established or not. And we have so some of these models have already been studied, and we are trying to characterize some new mutant transgenes as well. So the talk today that I am going to focus on is going to be on stem cell aging, and I'm going to focus on the intestinal system. The idea over here is to look at conserved regulation of stem cells across systems. That's why we are looking at 2 different stem cell systems in the lab. One is the intestine and another is the hematopoietic system. We also had a poster yesterday by my student Ujjaita, who works on the Drosophila model of cancer, cachexia, wherein we are trying to understand the blood cell mediated regulation of cachexia, how much the blood cells actively regulate this process of wasting in the organism. And I hope you might have visited her poster, and looking forward to feedback. So tissue homeostasis is achieved by maintaining a balance between stemness and differentiation, either of which, I mean, if there is a disbalance, it leads to a disease kind of scenario. And this happens either in a cancer scenario or degeneration or in an aging like scenario. So we are trying to understand how is this fine balance maintained. So now aging is one of the key factors which can affect this balance. There are a number of hallmarks of aging. It can lead to genomic instability. There is telomere attrition. There are epigenetic changes like Chandrima pointed out. There is proteostasis that is affected. There is nutrient sensing that is affected. The mitochondria gets affected, which Oishi spoke about. There are a number of factors that get affected as age progresses. So cellular and molecular functions are disrupted. What we are particularly interested in understanding is how are stem cells affected and how this impinges upon the overall organ and tissue homeostasis. So these are the changes that can specifically occur in stem cells, and these have been studied in the mammalian system. There could be DNA damage and mutations that can lead to apoptosis, wherein the stem cell population gets exhausted. There could be an alteration in differentiation. The microenvironment that maintains the stem cells could be affected, which can again lead to a disease-like scenario. There could be cellular senescence, loss of proteostasis, and mitochondrial dysfunction, which can affect the energy homeostasis in the organism. So the system that we are using to understand stem cell aging is the Drosophila intestinal system. This is the Drosophila midgut, and it is very similar to the small intestine in humans. The lineage of differentiation, the pattern of differentiation in the intestine is as follows, wherein there are intestinal stem cells that can self-renew. They get converted to the enteroblast population, which then further differentiates into the enteroendocrine population, which is the one that secretes the hormones in the gut, which is required for gut motility, sensation, and a number of factors, including nutrient absorption. Then the enterocytes are the large cells which are actually doing the absorptive function. So this is the lineage, and intestinal stem cells can also directly convert into the enteroendocrine cells, which is not represented here. But recent literature also suggests that there is another pathway wherein the ISCs can directly convert into the enteroendocrine cell. This is the way the cells are organized in the gut, and shown here is a picture which you will see in most of my slides in the picture, wherein the intestinal stem cells are marked by this antibody called as escargot. Enteroendocrine cells are marked by red in Prospero, and the large nuclei that you can see here are the enterocytes. So what we have done to study aging is we have genetically manipulated cellular aging, specifically in the intestinal stem cells and enteroblasts. So this we have done by either accelerating the aging or by reversing the aging in the stem cells. And we've done this also using chemical approaches, which is more broad stroke, because in genetic approach, you can specifically express something in a given cell type and look at its effect on the organ. But chemical is more broad stroke, but it could give you similar sort of effects. So for chemical induced aging, we've used hydroxyurea and paraquat. And for reversing aging, we have used rapamycin. And for accelerating aging genetically, we have used chronic inflammation approach, wherein we overexpress the NF-kappa B transcription factors, the NF-kappa B pathway factors, which can activate either the TOL and the IMD pathway, which are the inflammatory pathways in Drosophila, or we have activated the production of reactive oxygen species in order to induce aging. So now for reversing aging, what we've done is we've tried to make the cells younger by activating autophagy. We've used an autophagy effector, G8 in Drosophila, which would activate the process and it would ideally reverse age. And the other is the transcription factor FOXO, which has largely been looked at even in the case of flies, wherein it has been shown that if you express FOXO, overexpress FOXO in the entire organism, you can extend its lifespan. So these are the genetic tools and the chemical approaches that we've used, and you are going to see some of the data which points towards what happens in the stem cells and what happens to the organ when you do that. So the first part is what happens when you genetically drive these molecules specifically in the stem cell population. So like I said, we focused on chronic inflammation using the TOL and the IMD pathway and generation of ROS, wherein we have knocked down one of the electron transport chain components, which would help in generation of ROS, and it also leads to mitochondrial dysfunction. So the panel here on the left is the control. This is the chronic inflammation, and this is the ROS generation. We first looked at the cell proliferation in the midgut. Now, ideally, what happens is the green population here is, so the main thing to point out here is we use the GAL4 UAS technology. The GAL4 is tagged with a particular promoter wherein you express it in a particular cell type or in the entire organ. And the UAS is the construct which gets driven by the GAL4 protein. So the way we have driven it is specifically in the intestinal stem cells and the enteroblast population, which are marked here in green. That's why it's the escargot GAL4 GFP tag. And we've activated the TOL receptor, and here we have knocked down the negative regulator of the IMD pathway. So it would upregulate the pathway. And for generating ROS, we've knocked down the electron transport chain component. Now, what happens is when you look at cell proliferation, which we've marked using the phosphohistone H3, so mainly the ISC and the EB population is the one that proliferates. So if you go and look at the specifically the green cell population in either of these genotypes, what we see is that IMD pathway activation and ROS generation leads to an upregulation of cell proliferation in the midgut. TOL does not show a significant effect as compared to the wild type. Now, in terms of enteroendocrine differentiation, so we look, apart from proliferation, we looked at differentiation. What we see is that TOL and ROS generation is having a skewed differentiation. It leads to increased formation of these enteroendocrine cells, which could be harmful for the organism because a balance has to be maintained and the hormone producing cells have to be made in a particular amount. Otherwise, it could lead to enteroendocrine tumors in the gut. So what we see is that there are more of these enteroendocrine prospero-positive cells. We've quantitated this by looking at each image and counting them using the ImageJ tools. We've then looked at DNA damage using gamma H2X, which accumulates as a foci whenever there is DNA damage induction. And this is likely to happen in the aging scenario, as you must have also seen in my earlier slides. It's one of the hallmarks of aging. What we see is that, so the red staining that you're seeing over here, so what we've observed is that enterocytes have this background staining of gamma H2X, but when you actually sit and count the specific foci of gamma H2X, which are much smaller than how the enterocyte nuclei actually look quite bigger. But the ISC nuclei are smaller, they are in a cluster. And when you look at the gamma H2X foci in these cells, they seem to accumulate gamma H2X as compared to wild type. And that is common throughout all the three genotypes. So whenever we activate TOL, IMD, or when we generate ROS, we are seeing accumulation of DNA damage, specifically in this ISC and enteroblast population. Now what we looked at is gut integrity. So one of the markers of gut integrity is the tight junction protein coracle in the flies. So we looked at whether the junctions are getting affected. We are yet to look at the actual barrier integrity of the gut, which you can do using feeding the Drosophila larvae with a colored dye. And if the gut stains for the dye, it means that the integrity is intact. Whereas if the entire larvae gets colored with the dye, it would indicate that the gut has become leaky. So what we see for now is that the coracle or the tight junction expression lowers as compared to the wild type when you activate both TOL and IMD or when you generate ROS in the gut, which has been quantitated here by analyzing the localization and the expression of coracle protein. We then looked at a panel of DNA damage response genes and apoptosis genes using QRT for understanding whether the DNA damage response is getting induced and apoptotic gene cascade is getting induced. So we see a fairly differential profile. We do not see as a black and white scenario where all genes are getting upregulated. But what we see is that in the case of TOL, if you see here, ATM seems to be upregulated. So there is DNA damage response, but we do not see apoptosis happening in the TOL genotype. Whereas PERK leads to an upregulation of ATR, P53, CHEK1, CHEK2, and ROS. IMD activation has been known to activate the DNA damage response genes in other scenarios. Whereas we see activation of one of the apoptotic genes hid in the case of IMD. In the case of ROS, we see that CHEK1 and CHEK2 are getting upregulated, and REAPER, which is another apoptotic gene, is getting upregulated. Now, what we are planning to do is, this was done in the entire midgut. We are planning to look at specific apoptotic markers and do immunostaining to understand which is the cell type that's getting apoptosed, because that is important to understand in this particular scenario, especially when we are doing genetic modulation. So to summarize this part, what we see is that there is increased cell proliferation in the ISCEB cluster, especially in the IMD activation and ROS generation. There is overall increased DNA damage, and we see skewed differentiation wherein there is more amount of enteroendocrine cells formed. Now, when we chemically induce aging using HU or paraquat, which induces ROS, what we see is that HU is known to stall replication forks, and it is anti-proliferating, hydroxyurea. So we see that there is downregulation of H3P. We see less amount of H3P positive cells when you feed the larvae with hydroxyurea, but when you feed them with paraquat, we see an upregulation of proliferation, very similar to what we see when we induce ROS. So these concentrations have been standardized because it leads to lethality above these doses. So that has been optimized in the lab. We then looked at differentiation, and again here we see that HU is actually leading to a downregulation of this is the same slide. So in the case of Prospero-positive cells, we do not see an effect on in the HU treatment, but we see that in the paraquat treatment, it is leading to a downregulation. So this is what I was pointing at. There is a lot of cell autonomous and non-autonomous signaling happening here. So chemical approaches may not necessarily replicate what is happening when you specifically modulate certain genes in the genetic approach. So we are currently dissecting out what happens cell autonomously and non-autonomously and how are these cell types getting affected. But what we see is that there is upregulation of DNA damage in both the cases when we introduce HU as well as paraquat. We see gamma H2X positive foci. In fact, we also see that some of the ISC cells are getting exhausted. We see lesser number of ISC cells in the HU treated larvae. Again, another phenotype that was replicative in the case of HU and paraquat treatment was the coracal downregulation, which as compared to the control gut, when we treat it with HU or paraquat, we see downregulation of the tight junction protein coracal. We looked at the apoptotic and the DNA damage response profile. This we've done only in the hydroxyurea treated larvae, and we see that P53 is getting upregulated and HID is getting upregulated in this case. So now what happens if you reverse the clock? When you introduce anti-aging genes FOXO and ATG8 in the intestinal stem cells and enteroblasts. So like I said, if you activate autophagy, it's going to clear out the mess in the cell. It will make the cell younger. But it can also have other effects, which I'll come to when we looked at the actual phenotype in some of the cases. So when you activate autophagy with the help of ATG8, specifically in the ISC and EBs, it is going to accelerate, it is going to speed up autophagy, making the cell younger. In the case of FOXO, it has already been implicated in a number of, it is regulating energy metabolism, glucose metabolism, cell cycle arrest, DNA repair. It's involved in a number of processes, and it's an anti-aging, known anti-aging transcription factor. So when we do that, what we see is that you would expect that as compared to increased cell proliferation, you might have less proliferation in these cells, which is happening in the case of overexpression of FOXO, but increased expression of ATG8 leads to increased proliferation in the ISC and EB. What happens to differentiation? So in the case of differentiation, we see a reverse trend, which is exactly opposite as that of in the case of accelerated aging, wherein we see that enteroendocrine cells, which were showing skewed differentiation in the case of accelerated aging, now show a reverse trend, wherein the numbers of enteroendocrine cells become less as compared to the control. When we look at DNA damage, it's reduced in the case of autophagy, whereas in the case of FOXO, it's similar to the wild type. If you look at tight junction localization, FOXO shows a modest upregulation as compared to the wild type, but ATG8 looks similar. So now increased localization of tight junction and how that affects overall gut integrity is something not known in the field, and we are actually interested to do the dye assay and see what's exactly happening in these animals. When we look at the DNA damage response genes and apoptotic genes, again in the case of ATG8, where we saw increased proliferation, we are seeing that ATR is upregulated, and actually there are reports that CHEK1 regulator is described as a target of autophagy. So there are some reports pointing towards that, and we are seeing CHEK1 as well as the ATR, which is the activating kinase, being upregulated. And it also shows reaper upregulation in the case of ATG8. That's why it's important to actually look at which cells are getting apoptosed in this case. Whereas in the case of FOXO, we just see CHEK2 being modestly upregulated, and there is no upregulation of the apoptotic genes in the case of FOXO, which is again the opposite of that of chronic inflammation and ROS. So in this case, what we see is that there is differential cell proliferation. You can't make a blanket statement wherein proliferation is high in the case of aging and it's going down in the case of anti-aging. We see that in the case of some of the genes that we induce, but not in the case of autophagy. But there is definitely decreased DNA damage, and we do not see skewed differentiation that we observed in the case of accelerated aging. Now, when you treat them with rapamycin, rapamycin leads to decreased proliferation of the ISC-EB as compared to the control. When we look at the prospero-positive enteroendocrine cells, they do not show any effect. So there is no decrease as such in the case of enteroendocrine cells. DNA damage is lessened, so this is replicative again. very similar to what FOXO does. We see increased accumulation of gamma H2X positive foci in the ISC and EB. We also see increased localization of coracle at the membrane as compared to the control. So you also see an increased assembly of tight junction protein. It's not just expression of the protein that brings it together at the membrane or the junction. There are a number of proteins that associate together and form the tight junctions. So it would be interesting to look at how this junctional assembly is getting affected in this case. Whereas if you look at the DNA damage response genes and apoptosis genes, they're all down. So the question, overarching question after doing all this was, what is exactly happening when you're inducing these genes or you're activating anti-aging cascade in these cells? So what we did next is we took these methods. We generally, what we do is we dissect out the guts. It takes micro dissection and a lot of time to do that, but we took guts. We had to optimize the RNA isolation protocols for these and to have enough amount of RNA to do transcriptomics and good quality at that. So we did some transcriptomics analysis with the help of MedGenome, and analysis was done with the help of Nikhil Gardiwal at Actric, who's a bioinformatician. And we picked some common genes that are affected in the accelerated scenario and the decelerated scenario. And they fall into an array of array of molecular functions, starting from lipid homeostasis, adiposity regulation, nitric oxide synthase, to copper uptake, which is one of the main functions of the gut as well. So there are a number of things that one could look at. So we are now trying to actually boil down on which genes to look at and what function to look at, apart from actually delineating the cell autonomous and the non-autonomous scenario, because that's very common, especially when you're looking at the whole tissue level. There are cells that are signaling each other and dictating their phenotype. It's not just in isolation. It's talking to other cells in the tissue. To summarize my talk today, what we've done, and it might be redundant, but I would just like to go through it once. Genetically accelerating aging in the stem cell and the enteroblast population leads to increased cell proliferation, skewed differentiation, accumulation of DNA damage, leading to or resulting in an upregulation of the apoptotic gene cascade. And this is not a blanket statement that I'm making for all the genes, but in most of the cases. Chemical approaches, like I said, have their own caveats. It's not specific, you're doing it in the entire organism. So there could be a lot of interorgan communication happening, but what we do see is that there is increase in DNA damage and apoptosis in the intestine. Genetically reversing the clock of aging leads to a differential cell proliferation profile again, but there is no accumulation of DNA damage, and we see that the enteroendocrine cells decrease in number as compared to the accelerated aging scenario, which is also replicated in the case of rapamycin, wherein we see decreased DNA damage and apoptosis. Interesting phenotype for us, like I said, was the cortical localization of the tight junction molecule, which is called as actually septate junctions in the case of lice. Coracle, which is in the case of aging downregulated, and this is actually very replicative even in the chemical approach, as you have seen today. And transcriptomic analysis reveals a number of cell biological processes that are affected, and we are currently shortlisting genes to do our further mechanistic analysis. So these are the future directions, wherein first thing to do is to look at the cell autonomous and non-autonomous signaling. The good thing is we have GALFO drivers for each of these cells. So we can specifically modulate these genes in the enteroendocrine population or the enterocyte population, and then understand how the dynamics of the numbers of these cell population is getting affected. To validate the genes that stand out, we are going to pick some genes from this. Currently, we are looking at the gene ontology and gene enrichment analysis and doing some background research on that. We are also planning to do targeted metabolomic analysis, because there are a number of metabolites that have been reported to be affected in this scenario, and gut is especially important to look at for this particular analysis. Especially amino acids get up and down in the aging and the anti-aging scenario. So this we are doing in collaboration with Dr. Rakesh Joshi at NCL. And then finally understanding, because this is happening at the cellular level, we are not expressing these genes in the entire organism. So what happens if you age a stem cell of a particular organ? Is it affecting the entire lifespan of the organism? So we are currently doing these lifespan assays, which take some time, and you need statistical numbers, like in the case of whenever you're doing survival analysis in patients and stuff, you need the N number to be high. So currently we are doing that to and we want to induce it in the adult midgut to see what is exactly happening. So the time window has to be controlled in such a way to induce these genes. This is my group, and the work was spearheaded by Saraswati Pillai, who was helped with Grishma and Atharva. And this is the lab currently. My student, PhD student Ujjaita, is here with me today, and she had a poster on cancer cachexia. She's my very first student. Hargobind Khurana, IYBA, and Ramlinga Swami, SRG grant for the funding, and ACTRIC for gracious funding the intramural funds. Homi Baba National Institute, and I'm open for collaborations and interactions with other people. Thank you.
Yeah, the talk is now open for discussion.
Yeah, I have a question, Professor. See, this rapamycin-induced reverse aging. I was wondering if this could be extended to the mammalian cells. Have you looked at that possibility?
Currently, we are not doing that.
No,
But
I mean human cells rather.
yeah. One could study that in the human
Because
cells.
this
But
rapamycin has something to do with this mTOR pathway.
Yes.
Have you looked at the mTOR homologs in Drosophila? And are you looking at those possibilities also?
we will be looking at that when we do the mechanistic analysis. But the point here is, since we are doing it in the stem cell population, a good
Okay.
model, if we want to translate it in the mammalian system, would be to look at the stem cell population.
Stem cell population. Okay.
Yeah.
That would. Yeah. Thank you.
Wonderful beginning, Rohan, for your future. Um, I have a very, I guess it's quite naive actually. I don't know the system in Drosophila, but in C. elegans, I guess there are studies where you can take these stem cells and you can actually generate a gut-like thing in vitro, right? Where they kind of divide and then they
Right.
expand to form the tubes.
Yeah.
So some studies are most likely there. Now, my question is that these aged stem cells, do they retain that capacity
Of
to
self-renewal?
of self-renewal to form those guts, actually? Because, yeah.
Yeah, I mean, proliferation is one way of looking at self-renewal in vivo, but I'm not quite sure if, because no one has done it, and I doubt if these cells can actually be, dissected out and mechanically disrupted or enzymatically and go into culture and actually divide and form the organ, which no one has done so
It's
far.
not C. elegans, I guess it's zebrafish.
Yeah.
Yes, most likely
Okay.
zebrafish. But
Yeah, in culture, I mean, people have tried culturing the blood cells, which do quite well. In fact, there's also a cell line, S2 cells, which people use for their studies. But there are no reports of any of the organ in fly where the cells have been disrupted and they've been able to grow even for a day or two. But there are live imaging studies, so one could have tagged reporters or something where one could look at live imaging, because if you culture them in the cell culture media with some insulin in there, you're able to image them even for 24 hours. So that is possible to do. So if we could look at some of these tagged reporters, then that kind of studies can be done, wherein you can specifically look at whether these are self-renewing or not.
So on
That
a-
is your question, right?
Yes,
Whether
I
the
guess so.
self-renewal
Yeah.
is.
And on a more philosophical note, so these are the cells. The gut is one place where there is continuous renewal of cells, right?
Yes.
Still, you find like there are various populations of stem cells. One would be the younger ones versus the older
Yes.
ones.
Yes.
So yeah.
That's why it's interesting to see
Yeah,
how they are
absolutely.
coexisting together and what is exactly.
Any other question? No. If there is no more question, please. Oh. Here's a question, Professor Ghosh.
Is a rapamycin. Rapamycin creams are available. Beauty.
Rapamycin what?
Cream.
Cream.
For this beauty.
Oh.
So how safe they are?
Something that I've not heard of ever.
You are not aware?
How
But
could
it is
this?
available for as a beauty product.
Okay. So what about that? Can they be used?
Yeah, I'm
No,
asking
that's
that.
He's
Yes, how
questioning.
safe they are.
He's
to use.
asking how safe they are.
No idea. I got to know now for a matter of fact that they exist. I wonder how
No,
expensive
when
they are.
it is a cream means it is used for actually anti-aging, na? Yeah. Okay. I just I wanted to know how safe they are. Yeah.
I have
Thank
no
you.
idea.
Thank you. Thank you.
Organ transplant. So they have medical use, so should be safe enough. But this topical application,
I doubt.
I'm
Yeah.
hearing also first time.
Okay.
Let me ask Professor Kos, have you ever tried that cream? That's why you asked. Planning to.
Okay. Please join me to thank the speaker for wonderful. With this, this session is over. I sincerely thank all the speakers for giving wonderful talks and keeping time correctly. So thank you all the speakers, and I thank the organizer also for giving me this opportunity to conduct this session. Thank you.
So now I request the session chair to kindly present a memento to Professor Rohan. Thank you, sir. I also request to Professor Abhijit De to kindly come onto the stage and present a memento to Professor Maiti. Thank you, sir. So now let us proceed for lunch, which will be followed by the poster session afterwards. So I request to all the participants to display their posters if they have not done so.